Good morning, everyone. Let's go ahead and get started. My name is Campbell Ingram. I'm the Executive Officer of the Sacramento-San Joaquin Delta Conservancy, and it's my pleasure to welcome you all to the Delta Invasive Species Symposium, co-sponsored by the Delta Stewardship Council, the Delta Conservancy, and the Delta Interagency Invasive Species Coordination Team, also known as the DISC. The DISC team works to foster communication and collaboration among groups working on invasives in the Delta, and today's symposium is one of the ways the DISC team works towards this aim. You cannot overstate the significance of invasive species in the Delta and how they profoundly affect everything that we do, and that they're a persistent and very dynamic problem. So I want to take this opportunity to thank the DISC team for their leadership and thank them for their efforts to coordinate this symposium. And also want to thank the Stewardship Council's meeting support staff for helping to run the webinar today, and hopefully can deal with any technical issues that we experience. For the last 8 years, the Delta Invasive Species Symposium has been a forum for Delta managers and researchers, decision makers, and stakeholders to meet, share, and synthesize information and communicate best practices and lessons learned. In 2015, the symposium focused on invasive aquatic vegetation. In 2017, the focus was tools and actions to meet current needs and prepare for future challenges. And in 2019, the focus was on remote sensing applications for management. This year's symposium is going to focus on early detection and rapid response to invasive species. So again, I just want to thank everyone for being here and welcome you all. And now I want to pass it on to Dr. Laura Larson, the Delta lead scientist, to offer some opening remarks. Thank you, Laura.
Thank you, Campbell. And I am pleased to be here with you all. Hopefully you can see my slide now. But on behalf of the Delta Science Program, I would like to welcome you to the biennial DISC Symposium. As Campbell Ingram said, I am Laura Larson, the Delta lead scientist. Invasive species lie at the heart of management of the Delta's ecological resources. With at least 185 non-native species currently present, the Delta is one of the world's most invaded estuaries. Some of these invasive species, like largemouth and smallmouth bass, have been in the Delta for over a century. These invasive species have effects that permeate a large web of functional connections, not just to other species, but even to the physical environment. Most of us are aware of how the invasion and proliferation of the overbite clam around 1985 cleared the water column and caused a precipitous decline in the estuarine food supply, one of the factors likely related to pelagic organism decline. Clearing of the water column also created openings for invasion of non-native submersed aquatic vegetation, which now harbors non-native predators of Chinook salmon and other managed native fishes. This submersed aquatic vegetation is changing the turbidity of the Delta at large, according to work by Aaron Hester, and has contributed more to a step decline in turbidity than changing inputs of sediment to the system. Work published just this year by Jessica Lacey, Judith Drexler, Shruti Khanna, Maddie Foster-Martinez, and Rachel Allen has revealed that aquatic weeds are so successful at sequestering sediment that they may prevent sediment from making it up onto the marsh platform, thereby endangering the persistence of marshes in the face of sea level rise. And this does not even begin to get into the massive impact that aquatic weeds have had on recreational use of the Delta. The impact of invasive species has been phenomenal. In a 2021 paper in Science of the Total Environment, Crystal Ornelas et al. Calculated that in North America alone, control and damage costs of invasive species have amounted to over $1 trillion between 1960 and 2017. Here in the Delta, the Division of Boating and Waterways budget for control of invasive aquatic weeds has exceeded $12.5 million since 2015. The economic and ecological impact of invasive species make them a top management priority. To this end, in 2013, the Delta Interagency Invasive Species Coordination Team, or DISC, was formed to foster communication and collaboration among California state agencies and other agencies with a stake in detecting, preventing, and managing invasive species and restoring invaded habitats in the Delta. As outlined in the Invasive Species Coordination Framework, the goals of the DISC team are to establish a framework for strategic planning, coordinated implementation, education and outreach, data management, research needs, and funding. The work of this group, as well as widespread recognition of the problem, have ensured that invasive species management has been recognized in key strategic documents and calls to action. For starters, 2 chapters of the draft 2022 State of Bay Delta Science, led by Maggie Chrisman and Louise Conrad, are focused on invasive aquatic plant ecology and control, respectively, and that will be coming out in 2022. The Independent Science Board also came out with its invasive species review in 2021, which you will hear about from Steve Brandt momentarily. And then in the draft 2022 to 2026 Science Action Agenda, Action 3D calls for evaluating the costs and benefits of different strategies for minimizing introduction and spread of invasive species and informing early detection and rapid response, or EDRR, strategies. Meanwhile, in the draft 2022 Estuary Blueprint, Action 9 focuses on minimizing the impact of invasive species through prevention, early detection, rapid response, eradication, and control. It specifically includes tasks for creating a rapid response fund, funds for monitoring and/or mapping, and developing EDRR frameworks. Additionally, Action 12 of the Water Resilience Portfolio is focused on curbing invasive species altering California's waterways, including supporting programs that prevent, detect, and manage invasive species, and developing risk assessments, which encompasses EDRR programs. So what is this EDRR that is the focus of all of these actions and of our symposium today? Well, as Reeser et al. Summarized in a 2020 issue in the journal Biological Invasions, which is focused specifically on EDRR, there are many working definitions out there. Those authors attempted to synthesize those definitions, and they refer to EDRR as a guiding principle for minimizing the impact of invasive species in an expedited yet effective and cost-efficient manner, whereby detection is the process of observing and documenting invasive species, while response is the process of reacting to detection once the organism has been identified and response options assessed. This oft-used invasion curve that you see on the right plots the area infested as a function of time, with slow growth in the colonization phase, rapid growth and aerial expansion in the establishment phase, and relatively slow growth but pervasive occurrence in the spread phase. Control costs also follow this same curve. So it is clear that once prevention has failed, eradicating or controlling the species in the slow growth phase is the most effective and cost efficient, making this the window for EDRR efforts before the species becomes established. But there are many challenges associated with EDRR. Given practical constraints, where do you monitor for newcomer species and what do you monitor for? And given several new and possibly not so new invaders, which do you prioritize for control? Here is where I would argue it becomes absolutely essential to have a systems perspective or a conceptual model of the ecosystem drivers that limit species and the functional role that the species plays within the ecosystem in order to anticipate possible tipping points or other complex behaviors. Let me illustrate with an instructive example, which is that of Egeria densa, the Brazilian waterweed. Chrisman et al. Write in their draft SBDS chapter that Egeria densa was originally introduced to the Delta in 1946, but it remained in the slow growth phase for almost 50 years, not really taking hold and expanding until the 1990s. Now, it might have been the invasion of the overbite clam and subsequent clearing of the water column that opened a niche for its expansion, effectively serving as a tipping point. Assuming that it was a low priority species for control because of its limited aerial extent for its first 50 years in the Delta, in this case, would have been a mistake. One challenge of EDRR is that of anticipating which species, even those that might seem under control now, may pass through a similar tipping point, perhaps because of new niches being opened up as a result of climate change or shifting habitat boundaries with sea level rise, which in turn requires solid conceptual models that can help us anticipate future niche occupancy. This systems level thinking is an important part of the horizon scanning that Reeser et al. Would argue is a key component of target analysis, this symbol, in a framework for performing EDRR. I would say that it is also one of the most challenging elements. But when done successfully, it could help us anticipate the types of environments where new niches ripe for invasion may become available and can help us focus monitoring efforts. This is what I meant by unknown yet foreseeable in my title. Systems thinking can also help in assessing risk of a species entering a rapid growth phase in the near future. This risk assessment feasibility screening is the other element of the EDRR framework that I would argue is equally challenging, particularly the feasibility screening for treatment. Treatment here may need to be a novel technology or novel application of existing technology, which likewise requires venturing into the unknown. Indeed, success in EDRR is going to require innovation, making this exciting work for fresh, creative thinkers. On the detection side, how do we detect potential invaders of uncertain identity? The 2022 draft estuary blueprint calls specifically for developing new early detection tools using eDNA, adopting a technology that seems poised to revolutionize the field of environmental sensing. For many invasive species in our tidal environment, innovative new treatment strategies are also needed, as called for in Action 5A of the Draft Science Action Agenda. In her draft 2022 SBDS chapter, Louise Conrad and co-authors write that tidal flows often render aquatic weed treatments that work well in lacustrine environments, all but ineffective in the lotic environment of the Delta. So wrapping up, I'm going to leave you today with 3 distinct challenges as you think through an EDRR framework for the Delta. The first is to approach the challenge of horizon scanning and risk assessment from a systems perspective wherever possible. Second, exercise innovation and creativity. And third, think of how EDRR efforts may advance other science needs in the Delta. Now, number 3 here was inspired by thinking about a point made by Blanco et al. In a 2019 paper written about ecosystem disservices, mainly that ecosystem disservices, such as invasive species, may be a more effective impetus for pro-conservation behavior than ecosystem services. I think about the Frank's Tract Futures project, motivated in no small part by problems of invasive aquatic weeds impeding recreational activity near Bethel Island, but which resulted in design of a wetland restoration strategy that would have ecological benefits ranging far beyond invasive species control. So I ask you today, how can the actions of an EDRR framework have multiple benefits? Perhaps it's the use of eDNA for detection that could also generate information about how native species are using different habitats, or perhaps it is a conceptual model that allows us to identify native species refugia in the face of climate change. Regardless, I look forward to seeing what comes out of this symposium and subsequent efforts. Thank you.
Great, thank you, Laurel. Thank you for that context and that overview, and for those 3 challenges to focus on. Appreciate that. Next up is Steve Brandt, Dr. Steve Brandt, an esteemed member of the Delta Independent Science Board. He's going to present on non-native species in a dynamic delta.
Thanks, Eric. Are you running my slides?
I am, sir. Give me one sec.
Okay, thanks. As most of you know, the Delta Independent Science Board has a legislative mandate to review all of the science in the Delta. Largely, we've been doing that through comprehensive reviews of important issues that span the entire Delta, as well as span a number of different agencies. Our latest review, published in May, was on the science of non-native species in a dynamic Delta. And I'm going to give you a brief overview of that, and I think our recommendations actually touch on many of the presentations being given throughout the day. I do want to stress in this presentation the importance of science, anticipation, and coordination. The next slide. I think you missed one there. Yes, clearly non-native species are important. And in fact, they're one of the greatest global threats to ecosystem integrity and one of the 5 drivers of ecosystem change. As Laura mentioned, the California Bay Delta is one of the most invaded estuaries in the world. Non-native species are a key component of the Delta Reform Act, and reducing their impact is a core strategy of the Delta Plan. More broadly, the non-native species actually threaten our ability to achieve the co-equal goals, particularly the ecosystem component of the goal. And we argue that they even provide a question of what the ecosystem actually is. Next slide. Our review goal is to better understand the scientific needs related to this issue. Clearly, that helps agencies prevent and management the threats of and consequences of non-native species. Our focus is Delta-wide, spans multiple agencies, and we are particularly looking at a science-based prioritization framework to make decisions. The next slide. Our findings are no surprise to everyone on this call. The science of invasive species is extensive. It spans 6 decades. They impact almost every facet of ecosystem service or ecosystem sustainability. Interestingly, the basic needs and technologies dealing with non-native species at an individual level are really similar across a number of different ecosystems. There's been a lot of work done on this. It's fairly well thought out. And science is needed at each point in the management decision process. The next slide. When we look specifically at the Delta, we all realize the Delta is a highly modified ecosystem. It's still changing, particularly with respect to climate change. And these changes will affect the vulnerability of the Delta to new invaders. And although invasive species and the science related to it is similar across a number of ecosystems, the Delta does have unique institutional arrangements, responsibilities, collaboration mechanisms, and funding structures to handle this issue. Next slide. Most of you know the 3 ingredients necessary for the establishment of a non-native species. Number 1, you need to find a way to get them into the system, a pathway or corridor that allows the species to enter. Secondly, there's got to be some sort of a match between the species' inherent requirements, habitat requirements, whether they be physical, chemical, or biological, and those of the receiving ecosystem. And then thirdly, not all species are created equal, and some species have inherent traits, whether it's their habitat requirements, physiological capabilities, or reproductive capabilities that make them more of a threat than other species. Next slide. Our overall recommendations was to encourage a more ecosystem level and forward-looking integrated approach to non-native species, and particularly consider climate change. We stress that science prioritization and stronger collaboration across disciplines, agencies, and stakeholders are critical to get ahead of the game. We have 7 specific recommendations, which I'll very briefly touch upon. Next slide. Our overall review highlights the importance of anticipation, getting ahead of invasions for prevention and mitigation, really the topic of today's symposium. Perhaps of all of the issues facing the Delta, it's the integration of science, monitoring, and management that is absolutely essential in order to deal with this issue. When we think about it, the first step in the process is a threat or risk assessment. And if it is high, then we institute prevention mechanisms. If they don't work, we look at eradication. If that doesn't work, we try to control the critter. And if that doesn't work, we deal with adapting to the new environment, the new species. Now, management decisions are in the boxes, whether or not to take management action, but the monitoring tells us whether or not we are successful in that management action or not, and science provides us the way to monitor it, a way to assess the threat impact, and a way to take preventive actions. So they are very highly integrated. Next slide. When you zero in on rapid response and eradication, clearly it's important to detect and respond rapidly at a very early stage in the invasive process. A science-based, species-specific rapid response plan is really required up front to eliminate a species from an ecosystem, and that requires a team that spans agencies and including citizens as well, so that proper protocols and rapid response protocols can be implemented. Next slide. I'll go through our individual recommendations very quickly. The first recommendation is that we need a spatially explicit food web model for the Delta. Many of the impacts that invasive species have on an ecosystem has to do with how they affect other species, largely through the food web or nutrient cycling. Having a food web model for the Delta would allow us to improve our understanding of non-native species currently in the Delta, but more importantly, to predict the potential impacts of possible new invaders and assess how those threats would be altered by changes in drivers such as climate change. A second recommendation was to, when we want to zero in on what are the specific scientific needs, we recommend conducting a series of focused workshops and syntheses, such as the one we're having right now. And I might add that many of these recommendations we make are really fairly well established in the Delta, and we're highlighting them. And the next slide. The next recommendation is to identify and prioritize new species that pose the greatest immediate and long-term threats. This prioritization list should be based on evaluation of both expected ecosystem and economic impacts of high-risk invaders. Clearly, some of those are obvious: Nutria, zebra mussels, and so forth. We know there are lists for California. We think that as a state, we think that maybe a more focused list specifically for the Delta might be of value. Next slide. One of our larger recommendations is we need to go beyond the individual species approach and begin to shift our focus when dealing with non-native species or invasive species at the ecosystem level. If you think about the diagram on the right, the species pool is really the composition of species in the ecosystem, which is undergoing often constant changes, new species being added and other species disappearing. Largely that's driven by the drivers of ecosystem change, climate change, for example, being one, but land use and habitat alterations. Though the species pool dynamics is an important component of what goes on in most ecosystems and the Delta, we need to ask ourselves, what are the Delta ecosystem goals in the context of a changing drivers and changing species pool? We also suggest that we need to look at include non-native species in the context of ecosystem restoration activities and goals. The next slide. Of course, we have to look at the drivers and how they affect the threat assessments or risk assessments for non-native species, particularly in the context of climate change. Climate change and other drivers affect both the pathways, the types of invaders, the susceptibility of the ecosystem to invasion, and overall what the ecosystem impact might be. The next slide. We recommend that, I'm going to read this out because every word is kind of important in this, develop a comprehensive multi-agency invasive species coordination and implementation plan with the assignment of responsibilities and authorities that span monitoring, rapid response, control, and science expertise. There's a lot of effort. DISC has done a lot of effort in this, but what we are suggesting is that a plan be coordinated and implemented with assignment of authorities. We also recommend number 7 there that there's a single go-to science source of expertise that has expertise on taxonomy, rapid response protocols, technology, and so forth that is properly authorized and funded. And the last slide is a summary of what our conclusions are. Clearly, science can be used to better predict, detect, coordinate, and adapt to non-native species, and science monitoring and management are tightly integrated throughout this process. Being proactive in our understanding and monitoring is critical. Anticipation is perhaps the most important word in terms of minimizing impact of invasions. And lastly, dealing with the surprise invasion of Nutria. You know, the critter on the right, it's not a neutrina, but we need to ask ourselves, what would happen if this critter showed up in the Delta? Who do you call? Who has the lead authority on it? For rapid response, for example, does it matter whether it's a bacteria, benthic critter, fish, or mammal? Who has the authority? Who can act quickly? Who has the funding to enable to eradicate this animal before it spreads? So with that, that's the end of my talk. So I'm happy to take any questions if there's time.
Thank you, Steve. Were we going to do questions now?
I think we unfortunately need to move along with the program. But people are able to put questions to Steve in the Q&A, so please do, and tag them to him so that he knows that those are directed his way.
Great, well, thank you, Steve. So now it's my pleasure to introduce Rachel Wigington. Rachel is the DIS team facilitator, and thank her very much for all her effort to bring all 130 plus of you together for this symposium. Rachel is an environmental scientist with the Delta Conservancy, and she's going to provide an introduction to the symposium.
Thanks so much, Campbell. Good morning, everyone, and welcome again to the 4th Biennial Delta Invasive Species Symposium. As Campbell said, my name is Rachel Wigington, and I'm the Senior Environmental Scientist at the Delta Conservancy, and I'm just here with a few thank yous and some logistical reminders to help symposium today run smoothly. So I'd like to take just a few moments up top to say some thank yous. I want to thank the communication staff at both the Conservancy and the Stewardship Council who helped to coordinate emails and social media posts that brought so many of you here today. Thank you to all of today's facilitators, moderators, and timekeepers. A heartfelt thank you from myself and the rest of the planning team to all of today's speakers. This symposium would literally be impossible without your generous contributions of time and intellect. And a special thanks goes out to the symposium planning team members, Erin Heyman from the Wildlife Conservation Board, Gina Darren from the Department of Water Resources, Elizabeth Versati from the Department of Fish and Wildlife, and Rachel Klopfenstein from the Delta Stewardship Council. Thanks to all of these folks for making this symposium today happen. Now, just a few quick reminders. First, please be aware, this is a reminder to the audience that the symposium today is being recorded, and we plan to make it available on YouTube in a few weeks. Next, as has already been pointed out, we're encouraging folks to use the Q&A box to ask questions. So when questions are put in the chat, it can be really easy for them to get missed, and we don't have a way of marking if they have been answered. So please just chat in the chat and drop your questions in the Q&A box. And then we have a symposium facilitator today who's specifically watching the Q&A box, and we'll make sure that those questions are directed to the right speakers and are read out if there's time for Q&A at the end of different talks today. A reminder that today's symposium will run till 2:30 p.m. with a 30-minute break for lunch from 12:30 to 1. And we've really tried to spice things up today with a panel discussion before lunch and an interactive feedback session at the end of the day. So we hope you enjoy it and learn a lot. And with that being said, I'm happy to turn over the mic to my colleague, Erin Heyman, to introduce and facilitate the first session.
Hey, Rachel, thanks very much. Yeah, so welcome everyone to session 1 of this symposium, where the focus is going to be on early detection, rapid response, lessons learned. So we're going to hear from a few different speakers who have experience with invasive species control, and invasive species detection in either other watersheds or sort of wider geographic areas than just the Delta. And to kind of try to figure out what we can learn from those different geographies and apply to our own geography, the Delta. The speakers are Stas Burgell, Executive Director of the National Invasive Species Council. We'll be talking about early detection and rapid response at a national perspective. Teresa Thom, the Regional Aquatic Invasive Species Coordinator for U.S. Fish and Wildlife Service, who will be talking about EDRR in the Columbia River Basin. And finally, David Kratville from the California Department of Food and Agriculture, who will be talking about Hydrilla, Nutria, and Noxious weeds. Just a few quick reminders. Again, as Rachel mentioned, this session is going to be recorded, And if you do have any questions for any of our speakers, please be sure to use the Q&A box. The speakers should each have their talk and then hopefully a section for Q&A. So we'll sort of go speaker by speaker when it comes to the questions. And with that, I would like to turn it over to our very first speaker, Stas, all yours.
Great, thank you, Aaron. I presume somebody's going to pull up my PowerPoint. Great. Well, first I would like to thank the organizers for this. My name is Stas Virgil. I'm the Executive Director for the National Invasive Species Council, and I'm going to provide a national perspective. But I think it's important to recognize that a quote unquote national perspective does not happen in isolation. It's intimately connected to work at the regional level, the state level, and certainly at the local level as well. So I think, as we're going through these discussions, it'll be very interesting to look at how those different levels connect. Next slide. What I wanted to do today was talk just very briefly about the council, so you kind of have my institutional background, then go through some general concepts. And a lot of what I'm going to be talking about today is of the conceptual nature. I think Laura really sort of helped tee it up very nicely for me. So I'll be building on some of the remarks that she made. Then the policy context or the policy hooks, particularly that we've been operating under. And then finally, the bulk of the time will be sort of looking at kind of the conceptual issues around an early detection and rapid response framework. Next slide. So for the council, it was established by executive order in 1999, and that was updated in 2016. We operate through management or work plans. More recently, those have been annual work plans. The council itself is composed of 12 federal departments and 4 White House offices. So really our niche is that kind of interagency communication and facilitation of activities. And with the council, we work at 2 levels. One level is at sort of a political level, working with high level, they could be political appointees or senior career officials within the departments for decision making, but then there's also a programmatic level. So we work with folks on a daily basis within our member agencies. Then the council itself and its activities are facilitated by a small staff, which I lead. And finally, we also have a non-federal advisory committee, a FACA committee called the Invasive Species Advisory Committee, which provides advice to the council on particular activities. Next slide. And so this slide just provides kind of a visual. You can see, the various departments that are involved. It spans a range of different issues. Our co-chairs are from the Departments of Interior, Agriculture, and Commerce. But then we bring in Departments of Defense, Homeland Security, Transportation, international issues with Department of State, as well as 4 White House offices, which provides coordination more broadly with administration priorities. And looking across the breadth of these agencies and departments, they have different missions with regards to invasive species. They have different capacities, different capabilities, all of which, if you just sort of bring it down to the issue of early detection and rapid response, influences the way that they engage. Next slide. So Laurel presented the invasion curve, so I won't go through explaining it, but if you can click one more time. When we're talking about early detection, rapid response, we're really looking at that first part of the curve and sort of thinking about what are those prevention measures? What are the key pathways that we think invasive species might be introduced, monitoring and using our detection techniques at that point. And if something is found, then engaging in rapid response and really that early sort of section of the eradication. Next slide. So again, to sort of make sure that we're all on the same page, I wanted to sort of quickly again reiterate the 2 key concepts in early detection and rapid response. But over the course of the presentation, I hope to be expanding on those a little bit more and sort of building up to a more developed conceptual framework. So early detection are those activities that survey for and verify the first presence of a non-native species in a location. before that species spreads so widely that it can sort of not be efficiently or effectively controlled or eradicated. Next slide. And then if we look at rapid response, those are the activities intended to eradicate, and ideally eradicate a species from a location, again, before it spreads so widely that that's no longer possible. We do recognize that in some cases, eradication may not be possible given availability of resources or management techniques and technologies, in which case we might be thinking about containment or longer term management. But again, you know, in the first order, the hope is eradication and removal of that species. Next slide. So at the national level, and I think there's probably analogies when you're thinking at the regional, state, or local level, some of the targets that we're thinking about when we're doing EDR include the following. So click the slide. So when we're thinking about newly arrived species, and in this case, newly arrived to the country, one example being the Asian giant hornet, which was, I guess earlier this spring was found in Washington State, and addressing again those species that are newly introduced to the territory under question. Click it again. So then we also have species that are already here. So if we're thinking in the US context, we've got invasive carp species in the Mississippi River Basin, but they're not yet in the Great Lakes. And so there's a lot of efforts, obviously, to do early detection, rapid response activities in the connections between the Great Lakes and the Mississippi River Basin. Click again. Then there are those species that we anticipate will arrive sooner or later. So an example of this is BSAL, which is a fungal pathogen that affects salamanders. It's not in the US, it's predominant in Europe and other parts of the globe. And it's recognized that it could likely be introduced through trade in salamanders or other issues. So that's a particular species where there's been significant efforts to look at the pathways for potential introduction and try to find mitigation and management measures to prevent its introduction. But still recognizing that we want to have sort of early detection processes to monitor for potential introduction. And click again. Finally, there are the unexpected arrivals, the things you weren't necessarily anticipating. And while zebra mussels are prominent in the US, particularly in the Great Lakes region, it was a surprise again earlier this year when they were found in imports of aquarium mossballs. And while those mossballs were really algae, it wasn't one of the pathways that we were anticipating to come through. And so that involved a rapid response after they were identified, both at the federal and national level in terms of imports, but also at the state level in terms of commodities that were already for sale within the US. Next slide. So two of the major policy hooks that we have from the council's perspective in working on early detection, rapid response, relate back to a White House council and a set of recommendations that it made in the context of climate change back in 2014. And that was to look at a national EDRR program. And particularly in that sense, sort of thinking about issues with rain shifts and other sort of climate related dynamics that could influence the introduction and spread of invasive species. And then the second executive order, which was focused on invasive species, which again was really to look at particularly national and federal agency capacities to do early detection and rapid response. Next slide. But that's not to say that there hadn't been previous work. There's a range of documents that have been developed at the federal level by interagency committees as well as departments, focusing on various aspects of EDRR. So invasive plants, aquatics, forest health threats, focus on the Great Lakes. I would like to just focus a little bit on that last document on the list, the Safeguarding America's Lands and Waters. And that was developed in response to that climate change task force and the recommendation for developing an EDRR program. And essentially, that document, the 2016 Safeguarding document, tries to set up a conceptual framework for EDRR. Next slide. And pulling from that the list of elements on the left side of the screen really sort of show a little bit more what that report, the 2016 report, focused on. And then on the right, you see the diagram, and it's sort of, again, we're building on that sort of conceptual notion of early detection, rapid response, putting in preparedness as well as rapid assessment. So preparedness is those plans, coordination networks, tools, training, and other resources that are necessary to make sure that early detection and rapid response can actually happen effectively and efficiently. And then there's the rapid assessment part, which Laurel also talked about, which is determining the distribution and the abundance of the species in question, the potential risks it poses, as well as what kind of management strategies do we have to eradicate it. And so again, we're seeing the evolution of this conceptual framework, and again, I'll continue to build on that in the coming slides. So next slide. In terms of sort of the council, and again, where we've been sort of tasked to work on this issue, we've been engaged in the area since 2016. At that time, it was again looking at sort of federal agency capacities. And then more recently, we've been focused on some of the rapid response dynamics. Next slide. So one of the major outputs of the work that we did from sort of 2016 up until 2019 was a series of assessments looking at different elements of the early detection rapid response kind of continuum, particularly in the context of federal capacity and capabilities. And Laurel mentioned the article by Reeser a couple of times. That's where this was drawn from. The list of articles here were the results of those assessments, which were compiled into a special issue of the Journal of Biological Invasions. It's available online, open access. There is a URL, I don't know if it's showing on your screen, where they can be downloaded. And it relates both to pulling out sort of the conceptual framework as well as a blueprint, but then gets into more detail in terms of background information frameworks, data sharing, development of watch lists, screening, target analysis, identification, taxonomy, some of the innovation questions that Laurel mentioned, legal authorities, and finally sort of the incident command system. One of the things, and kind of in retrospect, having gone through this effort and worked with both the federal agencies as well as non-federal experts in doing these reviews, is that it's definitely a little bit more biased towards the early detection side of things. And that subsequently influenced the work that we've been doing over the past couple of years, which has been more focused on the rapid response side of the equation. And so I'll get to that in a bit. Next slide. So the diagram on the right is, again, trying to evolve that broader conceptual framework for early detection rapid response. And what I want to do with the rest of my time is to dig into each of those sections in a little bit more detail. Now, with the idea of what a national EDR framework should be, I want to provide the caveat that this is a conceptual model where in many cases we're trying to identify how do we fill in the bits and pieces that will allow us to implement the various sort of sections of this conceptual model. And it's also in thinking about the responsibilities of federal agencies on the early detection, rapid response, and invasive species front. We're also looking across a number of different topic areas, which themselves have different kind of levels of capacity and capability. So on the left-hand side, if you look at plant and agricultural health, animal livestock health, as well as public health, we have fairly well-defined sets of authorities as well as resources to address those, basically through the U.S. Department of Agriculture, as well as the Department of Health and Human Services. There may be some arguments about the adequacy of those resources, but the framing and the roles of the agencies is fairly clear-cut. When we get to issues around natural areas, as well as wildlife and zoonotic diseases, we find the involvement of more agencies. In some cases, the authorities aren't as clear, or there are potential gaps. And there may also be sort of issues with coordination or disconnects with regards to how things are managed, whether it's sort of regionally, taxonomically, and the like. So As I go through the following slides, I'm going to be providing some indicative examples of activities that happen in the various parts of this flow diagram. I'm not trying to sort of represent it as a comprehensive system, but I think ultimately that's what we're trying to build to, is a more comprehensive system. Next slide. So starting sort of at the top with horizon scanning, and from a national perspective, we're thinking about that from species that may be introduced into the US, as well as species that are within the US that might be moved around to different regions. And you could probably sort of develop your relevant analogies, whether you're focused at the national level, the regional level, state, or local. So at the international level, we've got the Customs and Border Protection, as well as APHIS, which have their kind of targeting and horizon scanning systems. But more recently, we've found with US Geological Survey, as well as the Fish and Wildlife Service, that they're developing tools and analyses as well, including looking at the pathways for the trade of organisms, transportation pathway analyses, as well as looking at specific areas themselves, like the Gulf of Mexico. Now, these activities, which are, have the involvement of federal agencies, are also complemented by work at the state and regional level. I provide one example with Florida on the bottom, which is conducting an all taxa analysis. They're completing that, and that should be interesting both in terms of the results, but also the methodology that they're using. Next slide. So moving from horizon scanning and sort of identifying where there is the potential for a particular species to entering into a particular region, we move down to watch lists and target analysis. And this is really where we're applying our risk screening processes, our risk assessment processes. Both Fish and Wildlife Service, as well as the Animal Plant Health Inspection Service, have various risk assessment processes. Some of those flow into regulatory lists, like the not approved pending pest risk assessment list. Others are more sort of advisory and don't have regulatory status. But those can help inform watch lists and the corresponding activities and target analysis that you need to do in terms of where should we be directing our monitoring and detection activities. And I should also note that on the bottom, there are a range of decision support tools to help this. I think recently I heard somebody had cataloged over 170 different risk assessment type methodologies. There's also population modeling, a range of niche modeling involving sort of climate change. So there's a lot of tools out there, and in that sense, some of the issues might be sort of figuring out which ones are better or more effective, depending on your particular circumstances. Next slide. So from there, we move into kind of the detection, identification, and reporting phase of things. With detection, different federal agencies have different sort of programs and capacities. USDA has a very sort of well-recognized one with the Cooperative Agricultural Pest Survey that they cooperate with states on. US Geological Survey is standing up a biosurveillance network, which is going to look at a range of potential introductions in natural areas, as well as with regards to zoonoses. There's also more specific pathway type monitoring, including ballast water sampling. And then also there's the technologies which Laurel had mentioned in terms of, for example, the use of environmental DNA, whether that's in sort of on landscapes, in waters, as well as potentially sort of at border in terms of imports that are being inspected through customs. Once a species is found, it needs to be authoritatively identified so that we're all certain that that's a species in question. Here, APHIS has their own national identification system, as well as a plant diagnostic network. Other agencies may rely on universities or other experts to help with that identification process. And then finally, once you have that authoritative identification, who needs to know? In some cases, that information can be reported to a database, which then sends it out to a range of different folks. I think it was the final speaker mentioned, are we communicating that information to plant health directors, to extension agents, who needs to know, and sort of get information to develop the next steps in the response. Next slide. Then there's the risk screening and the actual response. And here a lot could be said, but on the risk screening side of things, again, it's sort of thinking about what's the species sort of abundance, what risks does it present, what are the possible management responses that we can take. USDA tries to preempt some of this by developing new pest response guidelines. So for those species where they think there is a likelihood of entry, they will have a set of protocols set up beforehand in terms of how they would respond. Here also, in terms of the actual response, within the federal family, there's a lot of focus on the use of the incident command system as a coordination structure. And then finally, some agencies also have their own invasive species response team. So the Park Service and the Fish and Wildlife Service have regionally based strike teams. The Animal Plant Health Inspection Service also has, I think, sort of their own national level teams. Next slide. So here, at the core of this hub, it really gets back to some of the preparedness issues that I had mentioned, and particularly the importance of having a rapid response plan or framework in place to begin with. So, who are the agencies that are going to be involved? What are their authorities? What are the information and data systems that support doing the kinds of risk assessments, understanding the techniques that are available, the recording of occurrence data, decision supports, making sure that there is communication across those different information and data systems, that they're interoperable. Agency roles and authorities is an area that we're looking at right now. We recognize that there are agencies that have a primary emergency authority. So if you think about a first introduction into the country, if it's a plant pest, that would come under the auspices of USDA. But then there are cases where you've got agencies that are responsible for managing landscapes, water, or other assets, like it could be parks, national forests, hydroelectric facilities managed by the Bureau of Reclamation, where that particular agency is going to have the lead responsibility if there are introductions into or affecting its assets. And then finally, there may be cases where a federal agency is not a leading is not the lead on a rapid response, but instead is partnering or supporting. Maybe it's a state that's taking the lead. So, is there availability to provide resources, whether that's funding, personnel, information, expertise? And then finally, the training that needs to underpin all of this work to make sure that a rapid response itself is effective. Next slide. And then kind of the last bit is the funds that kind of can make all of this happen. We've also been working on developing the criteria and considerations for a response fund. In that, I should sort of provide a caveat that there is no federal agency request or congressional appropriation for a response fund. However, even as we saw in the introductory slides, this is something that has been mentioned and recommended at local levels, state levels, as well as national levels. So we thought it would be useful to go through the intellectual exercise of trying to understand what are the elements that need to go into thinking about the formation of a fund, such that if the stars ever did align, we would be one step ahead of the game in terms of setting that up. And some of the issues that we're looking at are listed on the left, and we're also taking lessons learned from other sectors where there are emergency response funds, for example, with wildfires, oil spills, and the like. Next slide. So I want to finish up just by noting some of the challenges. First and foremost, the conceptual framework really doesn't get into the geographical or the institutional aspects of coordination around early detection rapid response. So that's something to keep in mind. Other challenges are taxonomic. Are you looking for a system that's trying to address all taxa, or something that's focused on aquatic organisms, zoonoses, or terrestrial plants? So there's how do we look at it taxonomically? And there's also the geographic questions. And I would argue there's a lot of strength in regional approaches, But if we're looking across different systems, and I just put 3 maps up here, the Aquatic Nuisance Species Task Force, there are regional panels, the National Plant Diagnostic Network, as well as the 12 interior regions, or 12 regions for the Department of the Interior. And all of those are slightly different. So regional coordination gets to be an issue there. There are also gaps in authorities, as well as differences across the capabilities and capacities of federal agencies. Coordination can also be an issue, both within agencies, for example, across federal agencies, but also with non-federal partners. And then finally, the question of resources. I already talked a little bit about the availability with regards to a rapid response fund, but there's also thinking about the flexibility in terms of use of those resources, the timing and any constraints in terms of when and how they can be spent, as well as the ability to transfer them, for example, from federal partners to non-federal partners. Next slide. So I think that's it. Again, I appreciate the opportunity to talk and provide kind of the national perspective that we've been thinking through. And again, I look forward to learning a lot about what's happening within the Delta. Thank you.
Thanks, Daz. That was awesome. We do have a few minutes for questions. So Gina, do you happen to see any questions? Are there any questions that have come in? Let's see, it does look like
I sorry, I have a one tiny laptop screen problem happening right now. There is a question in the chat, and there's a question in the Q&A. So this one first, approximately how much time between between detection and an actual response measure, maybe in the typical scenario?
Yeah, and that's interesting, because that's something that we struggled with in terms of basically how do you, when you're talking about rapid, how rapid is rapid? And ultimately it depends on the taxa, it depends on the context. So if you're dealing with a plant, that's going to be very different than an invasive fish. And so it's kind of relative to the particular species that you're looking at. Obviously, in the latter case, you want to be sort of a lot more responsive, particularly if it can sort of move to other water bodies, whereas with a plant, you arguably have a little bit more time to react. And so I think we found that kind of the timing between detection and response varies. And in a lot of our work, we try to make it clear that notion of rapid is really dependent on the particular species and its ability to spread, whether that's sort of naturally within an area, or if there are particular pathways or ways that humans could spread it even further that might be of concern.
Thank you. We have another question in the Q&A. This one is, do you know of any federal funding coming to states for invasive species early detection rapid response? Good
question. Yeah, I mean, certainly within the agriculture and health sectors, there are already some programs that help with that. One of the things looking forward with the recent clearance of the Infrastructure Investment and Jobs Act is that funds have been made available to Department of the Interior as well as Forest Service, I think $100 million each for invasive species control. So there's potential there. Additionally, the Department of Transportation, I think it received an appropriation for about $250 million, which would be spread over 5 years, which would be a direct pass through to states for working on rights of way. So you could feasibly look at that as a potential source of funds to deal with EDRR in those particular areas.
Thank you.
Awesome. If there are more questions, it's fantastic. We'll try to get them answered, but we are just about out of time. So going to move on to our next speaker. But I think we have a way of labeling questions and sort of sending them off to specific speakers so they can actually respond to them in the Q&A or in the chat. So if you did post a question and it has not been answered, hopefully it will be in the course of the symposium. So with that, it's time to move to our Thank you, Stas. That was great. And thank you for all the information. And our next speaker, Teresa, is going to be talking about EDRR in the Columbia River Basin. So Teresa, all yours.
All right, thank you so much, Aaron. We'll give Eric a minute to get my presentation up. Thank you. All right, so again, thank you all for the opportunity to share some experience in the Columbia River Basin. And again, my name is Teresa Thome, and I'm our Regional Aquatic Invasive Species Coordinator with the U.S. Fish and Wildlife Service. And while you have me speaking to you, I have to 100% acknowledge that I am merely just one part of a large team throughout our whole river basin in the Columbia River Basin. And it's a mix of all different federal, state, and tribal partners as well. So you have me as our representative. So next slide, please. Just to help orient you a little bit, the Columbia River Basin, it is a very large watershed spanning over 260,000 square miles, including both Canada and the United States, several states in the United States, Washington, Oregon, Idaho. And it's the second largest river by volume in the United States. And there's a long, long history of human use by our native peoples, as well as more industrial development with hydroelectric dams, several dams along the river, and just a long-term use of this incredible landscape. There's very unique geology spanning a very long time series, and it includes a mix of federal, tribal, private lands, including things like the Columbia Gorge National Scenic Area. And again, it's a very unique ecosystem with rich biodiversity. And it definitely acknowledged that there's a lot to lose as far as this incredible resource with the introduction of invasive species. Next slide. And as you heard earlier from Dr. Larson and Dr. Grant, the impacts of invasive species to the Delta, we see across the United States impacts from different species. And this, I've got a map here going of invasion from zebra and quagga mussels from their first introduction in the 1980s from ballast water coming into the Great Lakes region, and then the subsequent spread across the United States. And as we learned more about the genetics of this zebra mussels, we found that there's actually two, the zebra and the quagga mussels. So, as technologies were being developed, we could tell that this was more than just one species invasion. And this, as you can see, it's pretty aggressive, the spread. And this invasion really galvanized the Pacific Northwest, all the agencies, to develop a preparedness plan for rapid response. And again, we're in the prevention phase still of preventing invasive species, the zebra and quagga mussels coming into the river basin. So next slide. So we did have the ability to really focus on developing a plan to be prepared in the event that invasive mussels would be detected in the basin. And so again, this was happening in 2000, and the agencies becoming galvanized on preventing invasive drissonid mussels, and then working to develop a plan. It took a long time to be able to look at a plan and write a plan and develop who was going to be signing it. But it was signed in 2008, and it's been updated. Our plan is available online. If you find the westernais.org, that website, there's all kinds of information about the plan and other resources that have been compiled related to invasive mussels, our best management practices and protocols. And again, this response plan is just one part of our overall management in the basin, preventing invasive mussels. Next slide. And just this signature page, if any of you, I'm sure all of you are very familiar with how hard it is to get support, but we're very proud of this signature page of having the support from multiple states and agencies on agreeing that rapid response to invasive mussels in the basin is a priority. It's considered an emergency and something that needs immediate response. Next slide. And so we practice responding to an invasion of invasive mussels in the basin. And this is multiple agencies are involved here. These exercises are held across the basin once a year, at least. We've been able to do an annual exercise every year, mostly since 2007. And with COVID, we've been able to do virtual exercises. So we've had to be flexible in how we do this. And it really shows, by doing these response exercises, it shows that our partners are committed to dedicating staff, letting staff attend this, and working through tabletop exercises and the process of responding to invasive mussels. And every exercise that we've done, we have those available, a summary online on our rapid response tab on the westernais.org site. So you can see the different activities. And next slide. In case you don't get a chance to go and look through all the different exercises, here just is a summary of some of the different locations, and these have developed over time. So the different states looking at different habitats, different locations where we may have an invasion, and just going through, well, who responds first? Who shuts the area down? Where do we bring in hot wash equipment? Who is the point of contact for public engagement? Who writes the press release? Who is actually doing a response? Are we able to put chemicals in the water? What endangered species are present? How do we handle those different issues? How do we engage Canada? And so those are some of the things that we've been working through as we go through these exercises. Next slide. And so some of our lessons learned, this is again, just focused on these rapid response exercises. They have evolved over time. And for every event, we have after action recommendations. So if this had been a real exercise, what could we have done differently? What could have been more effective? And one of the things that in 2019, we were able to look at kind of a longer holistic view of all the different activities that have occurred related to these rapid response exercises, and sort of see, well, are we missing some pieces? Are there gaps? What are things that we could do better? We're constantly trying to make sure that we're going to be effective. And one of the things we did find is that these after action recommendations aren't always addressed. And sometimes we've had common mistakes that we've repeated year after year. And one of the solutions we've tried to recommend is that it'd be helpful to have team members. Since we do move around, like if one year we were in Idaho working, one year we're in Montana, one year we're in Oregon, to maybe have team members that have been involved with the previous year exercise, just to give a little bit more continuity into the response, so that we could start looking at some of these after action recommendations and moving forward. So we're definitely not perfect, and we're trying to do better, and at least we have the opportunity to practice. Next slide. And overall too, looking at our rapid response exercises, we have a shared vision for the Columbia River Basin, and it's focused on preventing invasive mussels. And if we do find invasive mussels, we all agree that it is an emergency and that rapid response is needed. We've benefited from consistent funding, not from any one agency or a huge amount of money, but it's just been consistent that these efforts and information sharing and conducting these exercises has been an important priority. And we've also benefited from relatively low employee turnover in our different agencies. And so, over time, we have many years of experience working together on this issue. We have very productive annual meetings. It sounds like that is very much the same for the Delta. And we have an incredible list of dedicated partners that are 100% willing and able to dedicate time and energy to this work. And we continually work on our communication internally within our respective agencies, across programs, across different agencies, and then also to the public. And we work very hard to be flexible and as adaptive as we can, especially with new techniques and tools like eDNA, which has been mentioned, having the ability to kind of up our game as far as early detection. Next slide. And also, we recognize, or we are recognizing that our rapid response plan is a plan, and we wanted to be a little bit more adaptable in how we share that information and having it available online. And so that was sort of the latest push, was to develop a more easily usable toolkit for our rapid response. And this is available on our website. Next slide. So we have this online toolkit, and it's really focused with our resource managers and providing as many tools online, going through the, even compliance, the different forms that we need, having an incident command system. So just an online toolkit to do our rapid response. Next slide. And we are, again, looking at what are the next steps for our rapid response plan. Since the development of the plan in 2008 and recent updates, new partners are now engaged. A lot of our states have really grown their invasive species management plans and response capabilities. And so we're looking at how do we change up our rapid response plan. If you can click a little bit more, there you go. Also engaging our tribal community, our tribal partners. They are definitely leaders in conservation, and we want to make sure that they also are as engaged as possible with our rapid response. Next slide. Oh, there we go. Yep, and again, how do we engage or continually work with our federal partners and other groups? Next slide. And then, do we need to expand our geographic scope? We are very focused on the Columbia River Basin, but we are looking at, is it appropriate to include additional geographic range? And then again, as we're talking about early detection, rapid response, an important component of rapid response is making sure that we're monitoring and having the capabilities to even detect. And so this is just a map kind of showcasing all the ongoing monitoring work for dracinid mussels in the basin. And all the different colored dots, those different colors represent our different agencies, so different contributions of this monitoring data. This is from 2018. You can just see that there's a lot of monitoring work happening in the region. Next slide. And this is a more updated, this is a slide from 2020, again, just showing the ongoing work with the basin delineated there in kind of a reddish color. But you can see there's ongoing detection, the monitoring. It's harder to fund monitoring work. And again, all of our agencies have agreed that this is important, and we're able to get a lot of monitoring done. We have standardized protocols, but we do have a lot. This is part of our, our annual meetings, have our monitoring forum to make sure that we know where we're going to be sampling in the coming year, or at the end of the year where we actually did sample, looking at potential gaps in our monitoring data. eDNA sampling has been, again, a new technology that we've been very excited to use, again, with caveats that, a detection of eDNA is not the same as a detection using plankton or veliger sampling. And so there is a wide diversity of sampling techniques that we are using, but again, you can just see the large scale effort that's going on for early detection. Next slide. Also, we recognized that an important part of response needs consultation should we be detecting invasive mussels where we have threatened and endangered species. And so we are trying to be proactive. If you click a couple times, there are a couple, maybe a little bit, 2 more times. There we go. So we recognize that our consultation, we wanted to make sure that in the event that we did have invasive mussels detected, we could go forward with a response. And we can do that through the emergency consultation process with the U.S. Fish and Wildlife Service. And so ahead of time, we've looked at the threatened and endangered species known to be present in the basin. We've summarized potential treatment options that we would use to respond to invasive mussels. And we have best management practices to help avoid or minimize impacts to listed species. So we've gone ahead and worked, this was a long time in coming and developing this manual. And there is understanding that, again, if we do find invasive dracinid mussels in the basin, that constitutes an emergency and emergency response. Next slide. As Stas mentioned already, he highlighted a lot of different policies and guidance at the federal level for early detection and rapid response. And more recently in 2019, the John Dingell Jr. Conservation Management and Recreation Act provided very specific guidance from Congress to the Department of Interior to work not only with federal partners, but also the Western Governors Association and Western states on rapid response to invasive mussels. Again, this is very focused on dracinid mussels, and a real focus from Congress to use the incident command system. So similar to what is done for emergency response for like a fire or some type of an emergency that way. Congress was saying that invasive mussels in the West should use the ICS system, and that coordination should be occurring for rapid response activities. And we were very thankful that our work in the Columbia Basin fits very much in line with this directive. And ICS is an interesting framework to use in responding to something like invasive species. And it does provide a very streamlined process. Next slide. And we were able to sort of test our rapid response to invasive mussels very recently in March of 2021. And already the question in the chat was, what is the response time to an invasion? And I can, Stas mentioned that it depends on the species. In this instance, we had a detection of invasive zebra mussels in Seattle, Washington in March of 2021. That was reported to us. And we initiated our rapid response for that detection. And we had our partners, this is not the Fish and Wildlife Service, but our state partners in Washington were on the ground within an hour of getting the report. So that was their response time, was very fast. So same day. The delay was actually the reporting. And so when the report came of these contaminated mossballs with zebra mussels, that was filed in February and we were notified in March. So there was several weeks of delay. So, you know, again, it's not so much the detection, it's also sharing that information and reporting it to the proper teams to be able to respond. So that's another part in the process. So again, the contaminated algae with zebra mussels, that constituted an emergency response for our Columbia River Basin team. And while the initial report was in Seattle, Washington, it was quickly realized that this was more than just a one location. And then it quickly escalated from not just a regional issue, but an international incident, since it did involve importation of contaminated product coming into the United States. And so there was both an international response as well as a domestic response, and a lot of multi-agency coordination, as well as coordination with our industry partners. So pet stores, the pet industry, as well as the horticultural industry. And so this has really been a really important incident that's helped motivate and engage a lot of our partners on at the industry level as well. And since we did do a Columbia River Basin response, we had a lot of communication about this. We did provide a summary of our effort for the Columbia River Basin team, and we finished that in August. There will be a national review or national response after action report, looking at more of the federal response, and hopefully that will be happening in the next year in 2022. But again, it really highlights our need for constant vigilance and looking at the different pathways that we receive invasive species. Next slide. So that's really the summary that I have. I know that was fast. I should have some time for questions. And again, I am in our Pacific Northwest region of the U.S. Fish and Wildlife Service, and I want to acknowledge my teammate, Dr. Cesar Blanco. He is basically my counterpart in your region, and both of us are definitely available as resources. And I can speak for Cesar that we're both very excited and willing to be engaged in your EDRR efforts. So thanks again for the opportunity to share some of our experiences in the Columbia River Basin.
Awesome. Thank you, Teresa. It was a very inspiring talk for the work done in the Columbia River Basin. Very impressive. So we definitely do have a few minutes for questions, which is fantastic. Gina, did any questions come in?
We do have a couple questions in the chat, and for a reminder for folks, if you have a question for a speaker, please please put it in the Q&A, which I think will be preserved for posterity. The first comment that came in was from Virginia. She said, as someone with ICS experience on oil spills, it does seem as though California OSPR folks would have good experience for the EDRR exercises since they have a natural resource element. Do you involve the oil spill response folks in your teams?
So is that for me, that do we use oil spill response for the Columbia River Basin. Yeah, so many of our staff are trained with ICS for oil spill response, and that translates very well into our rapid response for invasive species. Similar to the ICS training that many of our teams receive for fire response. And that's what's so the strength of the ICS framework is that it's a standardized process for dealing with an emergency or an initial incident where you have a very clear communication strategy, very clear hierarchy of information sharing, and that's, I think, a very beneficial process for dealing with a lot of logistics, a big area. So depending on the the detection, what is trying to happen, there's at least the ICS framework is a known quantity where you have a lot of unknowns as far as your terrain and your response to the invasive species.
Thank you. And we had another question come in the chat. If you find veligers, what are the treatment options given that they are plankton?
Yeah, so veligers, finding veligers, so for those who are unfamiliar, veligers are the larval dracinid mussels. So what's difficult about invasive mussels and zebra and quagga mussels is that they broadcast spawn, so the eggs and sperm can form the veligers, which become adult or can become mussels without needing a host fish, which is different than our native mussels actually have a component of having a host fish as part of their life cycle. And so finding veligers in the water column is a high, I mean, that's a huge risk since the invasive mussels, those could become actual adult mussels and attach to things. And so, raw water is considered, high risk. And if veligers are found, treatment options are very difficult. So again, that's why we are working so hard on prevention, is that, once we get into the, detection and finding these things, we start having really difficult solutions as far as treatment. But there are some chemicals that have copper in them, which again, they're very toxic to not only invasive species, but also our native species as well. And again, finding veligers in a location, the immediate response is trying to figure out, is this from contamination? Are the veligers actually very abundant in the water column? How big of an area is contaminated? And so understanding, what is what are we looking at? What's do we even have the ability to control it? And so finding veligers wouldn't necessarily trigger treatment until the known, the area that's known to be contaminated would be evaluated. So we'd see if we can even treat an area.
Sounds very challenging.
It is
one more.
That's why we focus on the prevention. All right, there's one more.
So related to prevention, possibly early detection. With heavy biomass, what is the best way to test for veligers in the water column? Is it eDNA and in 30 seconds or less?
Ooh, yeah, I mean, eDNA is been shown to be pretty effective. Again, it probably would depend on your situation, but yeah, eDNA, especially now that we've had a lot of time to really test, the markers and how how effective it is. That's really been a very important tool.
Awesome. Thank you, Teresa, both for the talk and for the perspectives on the questions. Again, if questions were not answered, we will shift them around and make sure they get to the right people, and so we can get answers to them. With that, we're right on schedule for our last speaker of this session. David, all yours, and Nutria and Noxious Weeds.
Fantastic. Can you all hear me okay?
Yes.
Awesome. So today, again, I'm David Kraftful. I'm a senior environmental scientist in the Integrated Pest Control branch at California Department of Food and Agriculture. And I'm going to focus on my experiences with CDFA's various eradication efforts. And I want to focus on the successes we've had, as well as the importance of coordination. And it was great, the previous two speakers, many of the items they've covered, you're going to hear me repeat them, but with a lot of boots on the ground experiences. But I'm going to talk about Hydrilla, Nutria, and Noxious Weeds. So I am the supervisor of the Hydrilla Eradication Program. I am also the statewide weed coordinator for CDFA and oversee our Noxious Weed grants, which go out to the county ag commissioner's offices. I am also the secretary of the Vertebrate Pest Control Research Advisory Committee, and I'm in charge of our vertebrate pest research program, and that's why I was involved in Nutria when it was initially found, but I'll cover that in a few minutes. As previous speakers have said, where our early detection really starts in California is at the borders, the border stations, and I'm sure everyone here has been asked for fruits and vegetables when they come in from out of state. The geography of California, we're very lucky in that we have these beautiful mountains that surround us, and the big old ocean and the desert, and that really keeps species from just being able to come across our borders. When I'm in the Western Weed Coordinators meetings, which we had 2 weeks ago, it's interesting to hear a lot of the sort of Western states, like Wyoming, Kansas, Colorado, those areas. The invasion of weeds is just, they're just watching the wind blow and waiting for it to get to them. And we have this system built up where we can stop things at the border. Also, though, unique to California, we have this ocean, which brings things on boats directly, all around the world. And also lots of international shipments, air shipments, as well as travel comes in. So we have pretty unique sources of invasion, and we work on all of those at CDFA. But focusing on Hydrilla, next slide, please. So, as I said, I'm the statewide weed coordinator, and we are defined in the Food and Ag Code that the Department of Food and Ag is the lead department in noxious weed management. And we're responsible for implementing this article in cooperation with the Secretary of Natural Resources Agency. So a lot of coordination goes on as my role as statewide weed coordinator, which I'll finish up with. But next slide. People often ask, why do we specifically work on Hydrilla, though, when there's various other aquatic weed problems in the state. So legislation in 1977 in the Food and Ag Code mandates that the department conduct a statewide survey program and eradicate where feasible. It makes it unlawful to produce, propagate, harvest, possess, sell, or distribute Hydrilla. This is one of the few plants. We have our noxious weed lists. We have our pest rating system at CDFA. There are various other weed rating lists from different entities within the state, but for it to take legal action in the state, we really have to have them on either our noxious weed list or have them go through the pest rating proposal process. If you go to the next slide, please. Hydrilla is one of the only pests in the state that is named in a state law. So that bottom one there, Food and Ag Code section 6048, Hydrilla is name by name, stating that the department will conduct a survey and detection program and then eradicate where feasible. This is literally the definition of EDRR. EDRR has been my whole job for the past 25 years, or however it's been now. So it's just eradication work to me. At any rate, this is why we focus on hydrilla. And if you go to the next slide, the reason that hydrilla is named by name in state law is because it's considered the world's worst weed. And the reason for that is its impacts to water conveyance and storage. So the photo on the left there is a pond that was down in, I believe it was Madera County. And you can see this is what hydrilla does. It just 100% fills the water column. States in the southeast that have hydrilla, anagarria, and some of these other weeds that we're concerned with in California and the Delta, they worry about hydrilla. They don't. There was a lake in Texas that had a problem with Agarria densa. Hydrilla came in, and I think it was within 5 years, hydrilla was the predominant weed. Agarria was like hard to find. It just completely replaced it. And that 85% reduction in water and conveyance storage, hydrilla was discovered in the Imperial Irrigation District and caused water to just overflow the banks. Deliveries were not being made downstream. You can't pump water through a mass of aquatic vegetation like that. As well, the storage, that pond had lost 85% of its capacity to hold water. So you have 85% biomass of plants and 15% of your storage capacity is actual water when you have these complete monoculture infestations. And then the photo on the right shows the big issue with hydrilla is the propagules. And so you see that pond is lined with plastic. That can be very effective for mini weeds, but hydrilla, because of its tubers, those tubers just sit in the soil waiting for moisture to get to them, and then they sprout and you have a new infestation. So it's very, very difficult to eradicate, as I will show you. Next slide. The other big issue why we work on it and is funding. And I know that many of these partners are probably on the symposium today, but this is the reason that they fund us, is because hydrilla is such a major threat to a state like California that lives and dies on the ability to store and move water around. So we get funding from the Division of Boating and Waterways Harbors and Watercraft Fund. We get funding from California Department of Water Resources, and we also get funding from U.S. Bureau of Reclamation. Next slide. So this is hydrilla. Any of you folks out there in the Delta that are familiar with Agarria, and if you've spoken to a bass fisherman while you're out there, lots of bass fishing television shows focus on the southeast part of the country, and they fish the hydrilla mats. And I've been told by 100 different bass fishermen that they were fishing the hydrilla beds in the Delta. Hydrilla has never, ever, ever been detected in the Delta. We've been surveying for it since the 1970s. We've never found it there. But this is what hydrilla looks like. The whorls of leaves have 5. The Agarria tends to have 4, and then LAD will have 3. Oftentimes those whorls pack together, and the numbers can be all over the place. But if it's consistently 5, it's likely hydrilla. At any rate, the issue with hydrilla and where hydrilla comes from is where they have monsoon rains, and there's these ephemeral water bodies that come and go over a short period of time. And so hydrilla has developed this tuber, subterranean tuber, that it shoves down into the hydrosoil, and it can sit there for years. We can dry out a pond and kill every aquatic plant. As soon as the water comes back, hydrilla comes back. We had some great control in some ponds during the drought years, and when the water level came up, we just had this fairy ring of hydrilla that had been high in dry tubers for many years. Next slide. So this is where hydrilla has grown in California since 1976 was the first detection. We have over 30 successful eradications. We currently have active projects in 3 counties. Lake County, Clear Lake is the biggest of them. And then we also have Yuba, Nevada County. But you can see California is conducive to growing hydrilla. There are 2 biotypes of hydrilla. There's actually a 3rd one was recently discovered in, I believe, the Connecticut River. But we have dioecious and monoecious. We have both biotypes here in California. We actually have both growing in Yuba County. Interesting, those must have been 2 different introductions within several miles of each other, but they got the 2 different biotypes. But we've been very successful in eradicating hydrilla, and it takes persistence and rapid response and early detection. So next slide. And this is what we, why we go out and try, this is what we try to avoid. This is what, when you miss a early infestation, the eradication efforts get much more invasive, expensive, and quite a few of them we wouldn't even have available to us today. But this is I believe this is Ellis Lake in Yuba County, where the initial detection of Hydrilla was in the state, and they completely drained it and did some pretty severe actions to get rid of it. Next slide. But this is what a lot of our work today looks like. And one of the previous speakers mentioned that a lot of the costs of eradication work come towards the end, and most of it is just looking. And when you're not finding anything, it gets harder and harder to maintain funding when you're looking for a needle in a haystack and you're not finding many needles. But this is what the Hydrilla program, the majority of our work is. You see that on the right there, that's what we call them grappling hooks. They're about 8 inches across. We make them ourselves out of baling wire and pipe. And the crews there at Clear Lake go out in the boats and they simply drag these around, and we are looking for individual single plants. Next slide. And if you're familiar with Clear Lake, it is the largest freshwater lake in the state of California. Lake Tahoe is bigger, but half of it's in Nevada. So this is it is a gigantic lake. And you can see these are the detections from 1994 through 2016. Hydrilla has grown literally all over the lake. It likes the shallows, like most aquatic weeds, but we have to survey the entire perimeter of the lake. We also do some deep water surveys, but we focus mainly in the perimeter of the lake. And one of the issues that I'm going to get into in a moment is, and the previous speaker talking about zebra quagga mussel brought this up, and it's the institutional knowledge and continuity when you're doing one of these projects. You can look at a GIS layer like this, and there's a lot of information in there, but squeezing the nuance out of it from year to year and maintaining a project and an eradication effort can be difficult as managers change. So next slide. This shows the number of finds. So these are individual plants that were found over the years. And previously our protocol was that after we had 3 years of no plant finds in a treatment area, we would remove that area from treatment. We assumed that the tuber bed would be worn out after 3 years, and it would just go into a survey mode. So we were still doing early detection and just looking for those individual plants. Like what I'm trying to point out here is early detection and rapid response. It's part of an ongoing eradication program. We can look at each of these little locations as an individual population. We consider it one in that giant lake, but the importance of early detection and response go down to a very fine level, even on an ongoing project. So you can see that in 2004, 5, and 6, there were no plant finds in Clear Lake, and we began removing areas from treatment. So go to the next slide. And here's just a graph over that time frame of the number of plant finds. And you see right there in the middle, 2003 through 2006, there was essentially no finds. Go to the next slide. And here is the areas that we had in treatment. As I said, our previous protocol was to remove areas after 3 years and no finds. And you can see after 2006 and in 2000, we did no treatments in the year 2006. So go to the next slide. And here's the two graphs over the top of each other. You can see our protocol was wrong. And this is just adaptive management. We looked at what we had done, and the tuber bed is long-lived. And this is the importance of, this is the definition of that invasion curve. If you let these plants with these long-lived propagules, you allow them to set down a seed bed, for lack of a better word, this is what you're faced with. You can't just go pop one plant out of the ground and consider it done. So we've now extended our protocol to 7 years minimum. We keep treating for 7 years after we find a plant before we remove that area from treatment. But next slide. However, CFA has a statewide trapping grid, and we use this across most of our invasive species responses. We use this for insects primarily is where it's used. But they're 1 mile square grids. They cover the entire state. All the counties are familiar with the system. All the insect trapping programs are familiar with this. And we're utilizing it in our other systems as well. So we've put the statewide trapping grid across the lake. Next slide. But square mile is pretty vague when it comes to chasing plants. So we further divided it into these 8x8 cells. Next slide. And this is what happens, and everyone probably has a map like this. The importance of mapping cannot be overstated, but you have these locations, and as 20 years go on, the nuances between those dots can really get lost. And sometimes there's 14 individual ones in one spot, or is this 14 years of singles? So if you go to the next slide, what I'm doing now is we're utilizing that statewide grid occurrence to identify spots. So what I've done is the yellow line across the top, you can see about every 5 years, one pops up there. So is that an area that's getting reintroductions? Is it an area that our treatments aren't working as well for some reason? But this just really helps, and I'm the 5th or 6th person in charge of this program. So the knowledge of those local areas has gone away as folks retire. But this is also early detection. We're out there with those dragging those hooks, and the crews are absolutely amazing that they can find a single hydrilla plant out in that giant lake. But then utilizing that information, and we do have a protocol, we rapidly go out and we treat with a contact herbicide when we do find a plant, and then it cycles back into our regular systemic treatments. Next slide. And here I'm just highlighting, that's the sort of thing that I look for. If there's something that was recurring for some reason, and then we look into local variables. Is it water flow, sediment type, something about the chemistry in the water, the depth, and see if we have to adjust our treatments in some way for that area. Next slide. Okay, next I'm going to transition into Nutria. So we do have a pest rating list that includes vertebrates. Nutria is an A-rated pest. As I said, I serve on the Pest Control Research Advisory Committee. And because of that, I had a history of the eradication in California. And that was why I was one of the ones notified when the first animal was detected here in California. So go to the next slide. So in late March of 2017, a wildlife services trapper was doing beaver damage management down in Gustine, California. And he caught what he thought was a beaver, and it turned out to be a Nutria. That was in late March. In April, we put together a meeting. I went down to Los Banos and met with various entities. I received that animal, and that's the picture. That's the first day. In the recently deceased one at the bottom there was the first animal trapped in California in many years. The other one that's been mounted, we call him Nutty. He's been in my office since the 1960s. And so I was bragging at one point, I was the only person in California that had possession of the 2 Nutria in the state. Unfortunately, there was many, many more of them. We've since learned out on the landscape. But this was the initial coordination effort. We got together, CDFW, CDFA, state parks, U.S. Fish and Wildlife Services, irrigation districts, the private hunting club, and initiated that conversation. CDFW ended up being the lead agency, and they've taken over and continue to do the response. But next slide. I luckily had found some files. So CDFA had a history. Nutria were legally farmed here for fur beginning in the 1940s. My department had the records of the permits for those farms, as well as a number of escapes that had happened. So by the 1970s, we had considered it eradicated, and approximately 300 animals have been removed from San Diego to Humboldt County. The big numbers were right down in the Los Banos area. generally down in there. But this was the historical information that we had. So next slide. And again, the role that my unit played at the CDFA was doing delimitation surveys. So we utilize that same statewide trapping grid, just as we do when there's an insect detection. And we sent out a series of cameras to delimit the extents of the population. And then my counterpart there, Derek, is hanging one of those cameras in an irrigation system. And go to the next slide. And this should have a video. And if you get that to play, this is what early detection and delayed response looks like. So hopefully it plays. Nope, not going to play. Well, at any rate, if you go back to that one, I believe, and the folks from CDFW can correct me if I'm wrong, I believe this was the first time that we went and detected an animal outside of that core area. And it was, for a guy like me to watch that nutria, stand there and watch it swim away down the San Joaquin River, was heartbreaking. But unfortunately, due to changing environmental requirements, we were not allowed to respond. I was, however, shortly after that, able to take CDFW back to the location, and we removed an animal within a couple hundred yards there. So we assume we got the same one, but videoing him swimming away, it was heartbreaking. Next. The other portion of my talk, I want to talk about the Noxious weed program. This is my history. I was hired in the late 90s. We used to have district weed biologists, and that map there on the left shows the final iteration of those in 2008. That sort of pinkish colored district in the middle that went from Big Sur to Lake Tahoe down to Death Valley. That was my chunk of the state I was responsible for. I had the absolute best job on earth. Unfortunately, I'm the only remaining member of this group, but everyone else retired or went out of state. But what we used to do at CDFA as district weed biologists was EDRR. There on the right-hand side, you can see a picture of my truck, and in front of it is a Scotch thistle. And this was what we did. We drove up and down the highways looking for individual plants. And again, with these long-lived seeds, Scotch thistle has like a 20-year seed bank. The seed can sit in the soil for 20 years waiting to germinate. But if you can pop this plant out before it sets seeds, you can eradicate that single plant population. We had hundreds and thousands of situations where we did this, and unfortunately, we didn't do a great job of recording all of those. We did such a good job that no one knew we were doing our job. That's like negative success because you weren't seeing these plants up and down the roads. Unfortunately, in 2010, this program got cut. Next slide. So I'm just going to give you some examples of what EDRR success looked like. Here's Iberian or purple star thistle. Again, we would drive around and we would find these single plant infestations. Next slide. But it goes way beyond that. This is a seedling of Iberian star thistle. And this is what we were out there looking for. And killing that little tiny seedling is immensely easier. Next slide. Or even a small rosette. And this is what your search image is like. Or go to the next slide. And they keep changing and changing and changing and getting bigger and bigger and more difficult to control. This is a biennial, so it's got quite a taproot now at this point. Next slide. But what I was hired to do, and I'm that dark-haired college kid on the right there, my whole job when I was first hired was I was at the other end of a shovel digging plants out of the ground. And that's what early detection rapid response looks like. And if you can stop the propagules from being formed, you can eradicate these tiny little infestations. Much harder to do in an aquatic situation because it's much harder to find individual plants. Next slide. And lastly, I'm going to talk a little bit. This is fertile Cape weed. Again, this is an example of a single plant that I popped out with a shovel. Avoid the next slide, which is what it looks like when it spreads throughout a vineyard. As they did their tillage, they were spreading the seeds up and down. And this required much harsher methods, many years to wear down that long-lived seedbed. That's where a failure of early detection is. So finally, next slide. So currently, what I am doing as the statewide weed coordinator, we entered a partnership with CalIPSI to do regional weed prioritization. We rank weed species by their occurrence compared to the pest rating lists. We went out to the county ag commissioners and wild weed management area partners and identified regional priorities. And so what we're working on with this is getting that coordination going again. That used to happen with this district weed biologist, talking to all these county ag commissioners across county lines. And then we're also developing an EDRR email notification for the priority weeds that we've identified in this region. And I'm out of time, but next slide, that's it. Here's my contact info. If you have a question about Hydrilla, about the noxious weeds, or the vertebrate pest program, feel free to contact me.
Fantastic. Thank you, David.
Sure.
Great talk. Gina, did any questions come in?
Not yet specifically for David's presentation, but a question did come in earlier that I want to ask you, David, to start weighing in on. And then I would also like to carry this question forward to the panel this afternoon on implementation challenges. And that was an early question from Doug Johnson about, given that invasives tend to flow downstream, how can invasive species management from the Delta connect to management upstream outside of the legal Delta? Any ideas?
That's a tough one. And it is in a flowing system, it's a big challenge. Defining, do you put your effort on stopping propagules that are moving, or do you put your effort on the source? And there's different ways to look at it. You can make the analogy of a wildfire. You know, spots or spot fires are really dangerous, and you work your way back to the core is one of the ways that they fight fires. It has to be taken on an individual basis, I think. But it's certainly both. It's a two-headed snake, you know, and both of those are tremendously important. And it's one of the huge challenges. If you focus all your efforts in one area, you're going to get bit somewhere else. And that was why CDFA did the delimitation work on Nutria initially. We were trying to set the border of the population, while CDFW focused on the control work in the core. But he hit the nail on the head. A flowing system like that is incredibly difficult.
Well, I personally thank you, and I hope I speak for everyone here. Thanks for keeping Hydrilla out of the Delta. And
I'm glad nobody's ever seen it, hopefully, unless you've been.
That is like a personal nightmare. And thanks for keeping Nutria mostly out of the Delta, and we're going to hear more about that later today too. So that is all I see in the chat and the Q&A, but folks, if you have more questions, go ahead and post those and we'll make sure they get answered. Thank you.
I'm actually gonna jump in with the end here and ask a question myself, and that is, you know, you talked about part of your job was driving around searching highways for those noxious weeds. Have you ever sort of deputized or done work to deputize other people, just like the common citizen, to report on sightings?
So reporting is different. And we are trying to work out how citizen science is a huge thing now. And we are trying to work out how to, you know, if it just came to me, I could get inundated with millions of reports from across the state. So we are working on how to integrate that into our early detection. How to verify, you know, is it a valid report? Does it need to be chased? But historically, we worked with the county ag commissioners, those district biologists did. And there are a lot of legal issues, just like you can't go, you know, control weeds in the Delta on your own. You can't just pull over on the side of a highway and start pulling weeds or spraying weeds. So there are other legal constraints that are in place, but we certainly would do tons of training. Just knowing what to look for is such a huge thing. Just having that search image of something different. It's hard to know what's going to come unless you are in one of those downstream situations where you're looking for the thing coming from upstream. It's really training people to have that looks out of place. I used to always tell people, if you've never seen it before, it's probably either a native, which is super rare, or it's an invasive. So it's just having, and then realizing who to contact. And the first location to go to is usually the county ag commissioner, because they're pretty well aware of what's going on in their county, and they have the pathway and the knowledge to get it easily to our diagnostics labs for a final identification determination.
Awesome. Well, thank you to all of our speakers in this first session. It's been a pleasure, been very, very informative. And with that, I'm going to transition to our second session, which is being facilitated by Emily.
Yeah, thanks, Aaron. Can everybody hear me okay? Yeah, okay. Yeah, thank you, Aaron, and thank you to all of the previous speakers for their really interesting talks on the lessons learned from implementing EDRR. And it's my pleasure to start session 2 with 4 talks highlighting the successes and setbacks in EDRR implementation, with topics ranging from Nutria eradication in California, aquatic invasive plants, to assessing and reducing risks of potential invasions in California ports. And so these talks will be followed by a panel discussion. And our speakers for today's session are Jared Barr, Senior Environmental Scientist, a specialist with the California Department of Fish and Wildlife, Jeffrey Caudill, Senior Environmental Scientist, Supervisory with the California Department of Parks and Recreation, Ron Vanderhoff, Vice President of the Orange County, California Native Plant Society, and a joint presentation by Jonathan Thompson and Lena Ceballos Asuna with the California State Lands Commission's Marine Invasive Species Program. And each speaker's presentation will be followed by a 5-minute question and answer session as with the previous session. And as a reminder, please put any questions that you have for the speakers in the Q&A box. All right, so jumping right in, our first speaker is Jared Barr with the California Department of Fish and Wildlife, who will be presenting on the state of Nutria eradication in California from 2017 to 2021. Jared, if you're ready.
Right on, yeah, thank you, Emily.
Mm-hmm.
Cool. So yeah, hey everyone, I'm Jared Barr. I'm a senior environmental scientist with California Department of Fish and Wildlife. Thank you all for coming to my talk. Today I'll be discussing the state of Nutria eradication in California from 2017 to the present. We've learned a lot along the way, so I think this will be a good chance to share our lessons learned and our successes with the program, with special attention to detection of these animals and responding rapidly when found. So I'm going to start off with some intro material. Apologies for the review for some folks. And also I've crammed a lot into this talk. So apologies if I speak kind of quickly. Okay, cool. Next slide, please. So nutria are a large semi-aquatic rodent. Adults range in body size between 1.5 to 2 feet in length, and they weigh between 15 to 25 pounds. They inhabit fresh and brackish waters, but they do prefer low to no flow permanent water. They're native to southern South America, and like David mentioned in the last talk, they were actually introduced to the US in the early 1900s for the fur trade. And then from there in California, feral populations established from escaped nutria from these farming operations. They were, however, declared eradicated in California, from California in the 1970s, and then randomly popped up again and were rediscovered in 2017 on a managed wetland in Merced County. And then between then and now, they have been detected in 6 additional counties. So Fresno, Mariposa, Madera, Stanislaus, San Joaquin, and Tuolumne. Next slide. So nutria are highly fecund. They're prolific breeders. They will reproduce early and often, and they're reproductive by about 4 to 6 months. Their average litter size is around 6, and they can have about 3 litters every 13 months. They live in social groups. They're very gregarious animals, social animals, and their home range size is roughly about 25 acres, but they can disperse long distances up to about 50 miles as the literature states. So let's take a look at the kind of damage they can cause to the environment. Next slide, please. So Nutria cause 2 main types of habitat damage, one of which is vegetation loss. They consume up to 1/4 of their weight each day, and they prefer the basal portion of emergent veg, which means they can destroy up to about 10 times the amount actually consumed. This creates severe erosion and conversion to open water. And that means extensive loss of plant cover, organic matter for soils, leading to conversion of emergent wetlands and healthy ecosystems to more open water systems. And that can also exacerbate, their presence can also exacerbate invasive plant infestations with that. For example, arundo, parrot feather, and water hyacinth can take over following Nutria presence. Next slide. So in addition to vegetation damage, Nutria also create burrows, and burrowing damages infrastructure and levee systems. These burrows can extend up to 150 feet into the bank. They're multiple, complex, and extensive. So when you have a Nutria infestation, you typically have multiple complex burrow systems. This also impacts streams and rivers, which causes severe erosion, loss of bank stability, and increased sedimentation. Given their fecundity and the damage to habitat and infrastructure, Nutria are a high priority invasive mammal. And there are a few ways in which we can detect these animals on the landscape. Next slide. So our primary methods for early detection include what we call cell assessments, and then also deploying and monitoring game cameras and platforms. So we've overlaid a 40-acre grid on the landscape, and we use grid cells to help track eradication progress. It gives us a kind of historical record of our effort. And this grid system, along with other methods used, have been adapted from the Chesapeake Bay Nutria Eradication Project. And that Chesapeake Bay project successfully eradicated Nutria populations from over 250,000 acres of habitat on the Delmarva Peninsula in Maryland. So these cell assessments specifically consist of comprehensive sign searching and classifying habitat. Game cameras and platforms also play a vital role in the detection of Nutria. These animals often haul out onto banks or otherwise dry land to feed, groom, and do a variety of daily activities. So platforms act as a powerful attractant for Nutria. This helps us capture images of animals if they're present. Trappers will typically bait these platforms with sweet potatoes or vegetation cuttings, and will often use scent lures like Nutria pelt and urine as additional attractants. Since Nutria are social animals, using scent of conspecifics is a very effective tactic. Cameras are also important for monitoring in the short as well as long term. We have stations set up not only in the core infested areas, but also in areas of suitable habitat where infestation status is unknown. Of course, to have a robust monitoring network and survey effort, resources, equipment, staff, and partnerships are vital and necessary. Next slide. So as with the detection of these animals, staff, supplies, resources, and partnerships are needed to also initiate a rapid response. Next slide. And there are a couple additional methods that for detection that will be increasingly valuable later in the project when low density populations make it harder to find individual nutria and remnant populations. So the Judas project, for example, we capture nutria, sterilize them, affix transmitters, and release these nutria back into the wild. And because nutria are highly social animals, these Judas nutria that we're tracking will then lead us to more localized remnant populations or individual animals, and allow us to kind of target our trapping efforts more effectively. We're still in the research and development phase with this project. We're currently evaluating collars and tags that can be deployed effectively into the field. And then scent detection dog teams were highly effective in the Chesapeake Bay project, and we will be using scent detection dog teams. Unfortunately, that has been a bit of a delay because we aimed to trial these dog teams in 2020 and 2021, but the COVID-19 pandemic put a delay on things for us. Let's take a look at the current known geographic extent of the infestation. Next slide. So this is the current extent as of December 5th of this year. The main takeaway from this slide and the previous few is that to really successfully detect nutria and respond appropriately, we need our resources to match or exceed what is necessary with this infestation. So we need a robust monitoring network of cameras and platforms, dedicated field staff to conduct surveys on the ground, and partnerships with agency and landowner stakeholders, which can allow for additional land access and to foster growth in the program. So with all this being said, let's kind of take a step back and look at the history of eradication efforts in California. Next slide. So the first detection, or sorry, rediscovery, was made in March of 2017. Once found, local staff immediately responded and began trapping. But as David mentioned, in his talk, it was quickly realized that the infestation was more widespread than initially thought. And after realizing the scope, an incident command system, or ICS, was initiated in March of 2018. And with that ICS, land access agreements were created, field protocols were begun, a lot of interagency coordination was done at that time, and then short and long-term funding, some short and long-term funding was secured at that time as well. The ICS consisted of about 40 partially redirected staff from CDFW, and it should be noted that between 2017 and 2019, we had limited staff availability and equipment. So detecting nutria and responding was sometimes a challenge. We all know EDRR is the ideal, but in the beginning stages with our projects, we did not have a lot of resources at our disposal to deal with the problem as effectively as we would have liked. However, in 2019, we did secure additional funding to create a dedicated eradication program. Next slide. So, as I mentioned, we did rely heavily on interagency collaboration in the early stages, and CDFA, DWR, USDA, and US Fish and Wildlife have been involved in the nutria response since the beginning and have helped out immensely where they can. Owing to state and federal grants and an appropriation of state general funds, we did secure temporary funding for a dedicated program. So our partnerships with the Delta Conservancy and DWR, USDA and CDFA and many others helped facilitate this. And in November 2019, we were able to establish and expand our dedicated operations. So through these grants, we also were able to procure equipment and materials. Especially helpful were additional field vehicles and monitoring equipment gained via Prop 1 grant funding through the Delta Conservancy. And then budget change proposals, or BCPs, also allowed us to put staff into permanent positions. And in the 2019 and 21 BCPs, we obtained permanent positions for our grant-funded field staff. So through these, we can retain our expertise in the program and in the field. And I think previous talks have mentioned, the need for institutional knowledge to remain in the program. And that's what we've been really happy about with these BCPs. So we also saw the federal Nutria Eradication and Control Act get reauthorized and expanded. So this is also a potential source of funding and resources for the next several years. This was largely due to lobbying and with help from different special interest groups, such as the California Ag Commissioners and Sealers Association. So with this increase in resources, we then were able to increase our effort on the ground. In November 2019, we really started ramping up the program. Next slide, please. So this graphic illustrates our increased survey effort over time and hints at our progress these last few years. It shows the number of cells surveyed and number of nutria taken until November 2021. Note that the grasslands ecological area is a large area of managed wetlands, which is the core of the nutria infestation. And our field staff transition back to the grasslands every year, which is demarcated on this chart. So survey effort is measured in number of cells surveyed, which is the blue line, and the number of nutria taken or removed is the red line. Number of cells where nutria were taken is the black line. Note also when the ICS was initiated and when the dedicated program was established, with an increase in resources in November of 2019, we increased our effort to detect animals and respond quickly. So let's quickly zoom out and look at our annual take and survey effort. Next slide. Okay, so the green bars here on this chart represent the number of distinct cells surveyed. So we've drastically increased our survey coverage over the years, with 2021 having over 4,000 cells surveyed, which equals out to about 183,000 acres. And the red bars illustrate the number of nutria taken. Notice that our nutria take dropped nearly by half between 2020 and 2021, even though our survey coverage increased. This is indicative that our efforts in the last couple of years have been successful. We'd like to continue to see sustained and growing survey effort and nutria take continue to decrease. So the chart on the left further illustrates our progress, showing the percent of surveyed cells with nutria taken and then average number of nutria taken per trapped cell. Both of these metrics increased for 2020 when we ramped up survey effort and then dropped for 2021. Next slide, please. So with that, we'd really like to thank all of our partners and collaborators throughout these past few years. We have so many people to thank, many of which I was not able to include on this slide, but thank you all. And with that, I can take any questions.
Thanks, Jared. That was great and really awesome to see the progress that you've made over in the recent years. Yeah, we have a couple of minutes for questions. Gina, are there any in the Q&A?
Yes, we do have one in the Q&A. The question is, the nutria detection map showed all detections being in areas south of Stockton. Have there been any detected upstream of that we know of?
So yeah, I'm looking at the map right now, and we do have a couple. We did have a couple lone detections near Stockton. Notably, we had a roaming male, we think was a roaming dispersing male at Rough and Ready Island in 2019. We caught that animal on camera one time and then have not detected it after that. We do most of our detections in San Joaquin County, I should mention, are near Manteca and Lathrop. So the the southern tip of San Joaquin County. And from off the top of my head, because I pulled the numbers last week, we've removed 110 animals as of last week from San Joaquin County, but we've only gotten lone here and there single detections near Stockton. So we haven't taken any animals out of there. We've only detected random, every once in a while, a couple detections.
Thank you.
Sure.
Great. Yeah, thanks again, Jared. I think it's about time that we transition to the next speaker. But again, if there's any other questions in the Q&A, then those will hopefully be answered later. So our next speaker is Jeffrey Caudill from the California Department of Parks and Recreation, who will be presenting on lessons learned from controlling aquatic invasive plants in the Delta, as well as alligator weed.
Well, thank you, Emily.
Yep.
We'll just wait a second for the slides to come up. I just want to thank everyone for the opportunity to speak at the symposium today and talk a little bit about our control program in the Delta and how we've helped try to manage the alligator weed. So again, I'm Jeffrey Caudill with the California Department of Parks and Recreation and the Division of Blooding and Waterways. Next slide, please. So quick overview, just a little bit of the background. I would expect that, or I would anticipate that most folks probably are aware of our program. So I'll go into just some quick details about it and our current EDRR process. Then I'd like to talk a little bit about the alligator weed in the Delta and finally lessons learned. And we picked alligator weed for kind of two reasons. One, it's the most recent invasive that has been identified in the Delta with our program that we are allowed to control. And second, we don't have to yet deal with anything like Hydrilla. So that for that, we can thank David Kretfeld and his program for that. Next slide, please. So again, we're the California Department of State Parks and the Division of Blooding and Waterways. Per the Harbors and Navigation Code, we are the lead agency for control of aquatic invasive plant species in the Delta and Sassoon Marsh. So it's a regulatory part. On top of it, we control for environment, public health, and economy. And for those reasons, we are a control program. And I'll talk a little bit more about that moving forward. Next slide, please. Thank you for that. We have a lot of regulatory requirements that we have to follow, in part because we use pesticides, and the pesticides we use are herbicides, which are pesticides that target plants. On top of it, there's a number of endangered species within our program area, which is the legal, river delta and Sassoon Marsh. And so I've got a list here of the regulatory requirements that we have to go through. These are programmatics, such as the biological opinions that are conducted every 5 years. We work with U.S. Fish and Wildlife through Section 7 consultation, along with consulting with the National Marine Fisheries Services. The NPDES permit is the Water Resources Control Board, and that's based on using pesticides in water. The Streambed Alteration Agreement is for mechanical harvesting. The EIR is a part of the CEQA and the ESA requirements that we have. Restricted materials notifications are based on one of the herbicides that we use, which is 2,4-D, and then risk assessments, which we work with CDFW and they work with their partners to determine if a plant is actually invasive, which allows us to control it. Next page, please. Thank you for that. So we use an integrated control strategy. We have mechanical harvesting, which is the top picture. We use a lot of herbicides, which is the bottom right picture. And we have Dr. Patrick Moran in the lower left picture with the USDA ARS. And he's helping us, him and his team help us with biocontrol, which is something that we hope works for our purposes for control in the Delta. A part of the challenge with that is, well, I should say the benefit is it's an ongoing control strategy that one can use because we don't have to, feed them or pay them or anything else. From a resource perspective, it can be very handy. The challenge is most of the biological control agents that we know of like warmer temperatures. And so where many of the invasive plants that we handle today with our program are easily adaptable to the climate out here in Northern California, the biocontrol agent's not as much. So it's a continuing process. Next page, please. Thank you. So for our current EDRR process, basically we would work by ourselves and with other agencies to identify an aquatic plant that could be invasive and or a nuisance, and would trigger the opportunity for us to conduct a risk assessment internally and provide that to CDFW. And again, I mentioned environment, economy, and public health are the triggers for when we would put together a risk assessment, and submit that. And that would be, again, prepared and sent to CDFW. We will also submit a notification to the federal services to let them know that there is an additional plant that we may be controlling. With alligatorweed, and I'll talk about that very shortly, our risk assessments can take 3 months or longer, depending on working with, again, CDFW, working with their partner agencies. A number of the invaders that we may submit could be native species, which makes it more challenging for them to go through their risk assessment process. So in our EDR process, one of the potential challenges that I'll briefly discuss later is we aren't able to just identify a species and go after it, even if other agencies throughout California have that opportunity. One of the slower challenges for us is it takes a little while for the process to allow us to respond. But once it's deemed invasive and we get the approvals from the federal services and CDFW, it is a plant we can immediately target. Next page, please. So a quick timeline of alligatorweed in the Delta. So we worked with USDA ARS, and Dr. Brenda Gruelle is one of the subject matter experts over in kind of the Sassoon Marsh area, and she had identified and notified us of alligatorweed in Montezuma Slough. And so at that time, we had given a heads up to CDFW and prepared a risk assessment so we could submit it as quickly as possible, realizing that it could be in the Delta already. But we want to make sure that we have the regulatory processes and approvals to be able to go after it. And so when we had submitted to the federal services and CDFW notifications, by March 1, we were able to go after and target this plant. So we had started putting together some collaboration guidelines for aquatic invasive plant control, and that's AIP. And we identified as many sites as we could find using our field pest control specialists and technicians to identify known locations, and our field scientists to go out there and verify them. We tried to map and treat all of the alligatorweed populations that we could find. The California Alligatorweed Work Group was formed in 2019, where we work with several different agencies. And at that point, we have since then really continued to conduct treatments, identify all the locations, map them, and work with citizen science and outreach to determine where these are. And that's kind of where we are today. Next slide, please. So just a little bit about alligatorweed. It's a perennial, which is aquatic and terrestrial. It's generally rooted in the sediment, and but it can spread into the open, deeper water as a part of dense mats, and that's something that I'm going to show in some of the pictures later. We've been told that the mats have been reported to be dense enough to hold the weight of a person, so it seems to be somewhat of a relatively robust plant, and it spreads through propagules instead of seeds. And one of the things we've noticed too is that it's got a bit of a hollow stem. So if you were to go after it in terms of trying to hand pick it, you have to be careful, or anything you break off is going to cause a new plant or a new area of infestation, which we have to be careful of. And as I mentioned earlier, as part of one of the challenges with biocontrol is that it tolerates cold weather. But not prolonged freezing. And then on top of it, a part of the potential reason it took longer to identify it, it looks somewhat similar at a distance to water primrose, which is one of our target invasive plants, with the difference of opposing leaves, which are a little more difficult to see again at a distance, and the flowering, which is white, not yellow like water primrose. The next number of slides, I'm going to show some locations and some pictures from 2018 that we had taken just to see what we were dealing with. If you'd like to go to the next slide, please. So this is the picture I showed up front. This is a close-up picture of alligatorweed that we had taken. Next slide, please. For time purposes, I have a number of pictures, but I just kind of want to jump through them very quickly. As of October 2018, we had mapped all the known locations and sightings that had been reported to us, and this is where we were at. So it suggests that either the plant can travel very quickly, or maybe it's been in the Delta and it just hasn't been noticed until the notification from USDA. And we also had a notification, as one of the pictures will show, again from CDFA, where I'll point that out. And I think that's where David Kretville showed us a location just right over by the Tower Bridge in Sacramento. Next slide, please. So a lot of these pictures are going to have the Delta map that we use to show our sites, and it's going to have a yellow area a yellow arrow that will show you where these are. So here's an example of a relatively large patch of alligatorweed. Next slide, please. We'll kind of jump through these quick, and some of these can be closer to the shoreline, which might make them a little more difficult to find based on how invaded the area might be with other plants as well, as many of these invasive plants don't necessarily make up the sole source of invasive plants in a specific site. Next slide, please. This is the picture right near Tower Bridge, where a couple of us gathered up after David Kretville notified us that there's alligatorweed not too far from our office. So next slide, please. All right, next slide. And we're just going to very quickly go through these due to lack of time. Next slide, please. This is where it's right up against the shoreline, as I mentioned. Next slide, please. And and most of the time when we get reports from the public for ease, they might call it water hyacinth or Aguirrea densa, which are the common invasive plants that we deal with. So sometimes when we get notifications of plant or aquatic plant control needs, we try and make sure that everyone in the field has the training to identify the plants, because this was a case where somebody just said, come get the water hyacinth. And we had ended up identifying it as alligatorweed. Next slide, please. And this is, again, a picture I'd mentioned where you see water hyacinth in the picture along with alligatorweed. Next, please. And you can slowly go through these until we get to the next couple of slides where I can talk very briefly. Next slide, please. And that's just a small site. So as of 2020, we ended up having a significant amount of additional detections, which make this plant relatively difficult to control for our program. And in part because we have a lot of different stakeholders within the Delta that kind of demand and require our attention. And we try and do the best that we can to control as much of these aquatic invasive plants as possible, while providing for navigational waterways for, say, the ports of Stockton and Sacramento, helping with boating access for recreational access at marinas and ski runs and things of that sort, helping out agriculture. And so a lot of you'll notice a number of these locations are also in the Sassoon Marsh as well. Next slide, please. So lessons learned, this is not something we can handle alone. So as a part of the early detection rapid response, if by others reaching out to us and helping us understand the nature of alligatorweed and its threat to the Delta, it allowed us to try and get after this plant relatively early. But it also lets us know that by collaborating with other subject matter experts and other agencies, we might have had a better chance if we had started getting after this before there were 20 to 30 known locations that we knew about after we started getting the risk assessment process going through. And again, the collaborations are key here, because we're a control program and not an eradication program. So as much as we would love to be able to aim at eradicating these plants, there are some limitations to our program. And so earlier notice would benefit our program. And so part of the lessons learned we've got is we will make sure that we are reaching out to all these other programs, and many of them are here represented on this symposium, to make sure we know what to start looking out for instead of the 9 aquatic plants that we have now. And we've also learned from past risk assessments that advanced notification to CDFW makes a big difference in order to get a quick turnaround time on the risk assessments, because if we just notify them, through an official request for a risk assessment, it doesn't give them a lot of time to prepare for that and work with their stakeholders as well. Next slide, please. So ongoing work. we continue to revisit these sites. We continue to document where they are for alligatorweed and do the best that we can on trying to treat them and control them, along with additional surveys based on the work that our field scientists attempt to do, based on limited resources and time that they have for that. We also try and make sure that we share any of the sightings with the other agencies that we work with, like in the Alligatorweed work group, for example. And I think, as others have pointed out, collaboration makes a big difference here, as going at it alone is not exactly the key to success here. And so that's something that we are continuing to work on, and we strive for improvement. Also, it'd be nice to find out where some of the source populations are located, at least within the Delta. However, it's spread quite a bit at this point. Next slide, please. And so if you've got any questions or comments, the easiest way to go about this is to reach out to our email address there. We get quite a few emails based on any kind of plant issues, reporting sightings. Otherwise, contacting anyone within our program works as well.
Thank you so much, Jeffrey. That was a great talk. And unfortunately, we don't have any time for questions, but it seems like there are some in the Q&A if you wanted to check those out and answer them. And yeah, we're going to move on to our next speaker right now, who is Ron Vanderhoff with the Orange County, California Native Plant Society, who will be discussing successes, failures, and lessons learned from an Orange County Emergent Invasive Plant Program.
Good morning, and thank you. Thanks for inviting me. I think I might be one of the few Southern California people here. So I don't know how I got in this. I'd like to work pretty quickly, but take a few minutes and talk about a program that we initiated here in Orange County that may have some value in the Delta. We initiated it in 2015, we went public in 2016. It's a program of the local Orange County chapter of the California Native Plant Society. One distinction in this, it is completely a voluntary program with zero funding. So a nice way to engage citizen science and such in some of the conversations that we're having this morning. So I'll outline the program, talk about successes and failures, and then a few lessons, and try to do that in 12 minutes. Next slide. When we initiated, we've been engaged in invasive plants forever in the CNPS chapter, but when we initiated this particular focus, that's pretty good. We wanted to understand where we could participate and be useful in the invasive plant world. And we wanted to avoid overlap. We wanted to accentuate our strengths in CNPS and our membership and not create duplication of effort. So we chose to identify that niche through assessing our strengths and weaknesses in the larger issue. Next slide. So a little bit about our strengths. This is actually our weaknesses here, but our strengths, we have expert knowledge of the Orange County plants. We're field botanists, many of us. We have an organizational structure. We have partnerships with all the land managers and agencies already, and a lot of volunteers and very little bureaucracy. Our challenges, which is this slide, is, as mentioned, we're not a funded organization. We don't have any grant money for this work. We're volunteer driven, which is very cyclic. We have no authorities. We're advice only, and lots of jurisdictional challenges, particularly land access, which will come up in a minute. Next slide. So we wanted to see where we could fit and again make a difference in that larger puzzle that was already occupied by various agencies and land managers and such. Next slide, please. So what we settled on back in the formation of the program was to pick a very specific niche and stay within that niche, and that is the early detection and facilitation of effective management of specific early emergent invasive weeds. So the key points there is it's very much about early detection. In fact, the earliest detection possible. We want to find that needle in the haystack. We want to get it when definitely we can eradicate rather than manage. And we wanted to facilitate that management, although we knew we were not a management organization. And we were specifically focused on what we are calling emergent invasive weeds, the very beginning of that invasion curve. So next slide. So that was our niche that we felt our strengths would occupy quite well. We needed to do a few things. So one of the first things we had to do was define to ourselves and to our partners what we were defining as an emergent invasive plant. And these are the very earliest occurrences within a region. They would have very limited distribution. We wanted to be sure the horse wasn't out of the barn very far. If it was, that would be for other groups and organizations. We needed to assess those plants for their potential for environmental challenges and their potential for invasiveness, ecological disruption. And there's some challenges inherent in that because these are new weeds in a new location. Some of that is speculative, and we had to use some modeling as best we could to assess those invasive and ecological concerns. Next slide. Another task right from the beginning was to assess those plants, those candidate species, and prioritize them. We just, again, just to remind that they had to be emergent, they had to be very small populations, not very far along the landscape. We solicited candidate plants. We put them through a rather rigorous data-driven scoring process. And we knew that we would have a dynamic list that would change annually. We didn't want something to be very long-lived. We wanted to review these and reassess them on an annual basis. We knew that species would be added and removed as we moved along in time. Next slide. I don't expect you to be able to read that detail, but just to demonstrate a little bit that we did want the assessments to be data-driven and not a bunch of land managers and botanists sitting around with a beer and deciding, you know, this is, put this plant on the list and not that plant. So we developed a protocol and an assessment algorithm that was rather data-based. We assessed abundance, populations within the county. We assessed distribution by looking at quarter quads and where those plants were distributed within the county. We assessed their ecological impact, a couple factors on that, their potential rate of spread, annuals versus perennials and such, trees and grasses and such, and a couple of other variables. Where we could, we used Calypsy data, which is rather data-driven. Where Calypsy data wasn't available, we created our own mirror assessments of those ecological impacts and rate of spreads based on our local expertise. The nature of new discoveries and new species is that often you don't have those assessments in place. So about half of the species that we're dealing with have no Calypsy assessments yet. They're too new, they're too emergent in the state. Next slide. To develop that knowledge base, we, as mentioned, relied on Calypsy. We relied on other online papers and research, mostly from our region, but from other Mediterranean climates, other unpublished data, and a large volume of local knowledge of field botanists, land managers, invasive plant experts in our region. Next slide. What do we do after that? We established a committee. Every project had to start with a committee. And next slide. the initial, well, actually the current committee, this isn't the initial committee. The current committee is a bunch of people there. We tried to include a diversity of people from different agencies and land management groups, as well as NGOs, and keep it a manageable size. Next slide. We created a website, next slide, which I will also mention toward the end here in successes and failures. We did this. We invested too much energy and wouldn't have done it quite the same. Next slide. There's more about the algorithm and so on. You can go to the next slide. And then we published our first priority list that distributed to all the land managers and people engaged in invasive management. Next slide. This happens to be the current list that we're operating with at the moment. It'll be revised for 2022, but we typically have maintained between about 15 and 18 taxa at any given time. And these may be common in parts of the state, but in our region, they're emergent, very emergent, they're very, very limited populations. Next slide. And we published a watch list as well, species that are around the county that we think are moving our way and may likely show up in time. Next slide. And you certainly can't read those, so you can go to the next slide. And then we also did some training. We created some resources, some tools, and a year or two later, we initiated some rather extensive training sessions for land managers and field people. Next slide. These are some of the resources. We have plant profiles that are distributed. Those are the larger documents for each one of the species. And these pocket ID cards have become very popular. They're laminated, and rangers and others can bring them out in the field and use them. Next slide. And the trainings have been quite popular. One of our expertise were really good botanists, and we're able to take parks people, Forest Service people, Caltrans, and NGOs and volunteers and so on. And we can do some pretty extensive EDRR training and talk about identification, reporting, and so on. Next slide. I'm going to roll through the last couple slides here. Keep going. So successes, these are just a few. We've had several first or second or so detections, several in the county, a couple in the state through our program, and have been able to turn that information over to the appropriate land managers to initiate eradication. Those are some of the species that have been high profile in our program. Next slide. We've also just raised awareness in general of the invasive plant issue, looking at CalFlora, which is probably the largest repository of this data. Prior to our program in the county, we had 3,000 Calypsi species records, and in the last 5 years, that number has jumped to 22,000. So just general invasiveness, not just emergence, has drastically increased. And of course, not all of this is from our program, but we've contributed to that. Next slide. So lots of detections, lots of new records, lots of people trained, some tools that are out there in use right now, and then lots of intangibles, building a community and reinforcing that invasive plant community, and really engaging a lot of citizen scientists into this issue and drawing them in. Next slide. If we could go back in time and do it again, what would we do? Well, not really a failure, but species have left the the barn. We've lost a few of them. They've moved past the emergent stage and they're off our list. They're into management programs now rather than eradication programs. And so they're out of our sphere. We spent too much energy on our website. Suggest less of that. There's other websites that can handle that. And as any volunteer people will know, sustaining volunteers is often challenging. And I think the last slide, maybe. Lessons, if I were to do it again, or there was a similar effort in the Delta, I would recommend find a niche, don't overlap, stay in that niche. That's very, very difficult to do because you want to get pulled in many directions, and find that niche where you can make a difference. Prioritize the weeds, provide resources, training. I'm going to jump to the last one, create a feedback loop, especially for volunteers. If a volunteer goes out and spends energy and turns in an observation and never hears anything, that volunteer isn't going to stay engaged very long. Have to have feedback. And then probably one of my lessons that I really, really always suggest is embrace one data sharing platform as much as possible. And I know that's got a lot of agencies listening here, and that's very, very difficult to do. But if everybody has data locked up, it makes the larger issue very, very difficult to deal with. And then learn the people. And do I have another slide? I do. I just want to thank everybody that's participated in this committee, and our co-founder in the chapter. And you all know Dr. Jutta Berger now up at CalLibC. She left us, but we're carrying on. She was one of the founders of this program in Orange County. Thank you very much. I think I'm over my time.
Thank you so much, Ron. I really liked the creation of the pocket identification guides, but great talk. Thank you. Yeah, we're gonna have to move on to our next speaker, our last set of speakers actually, which are Jonathan and Lena with the California State Lands Commission's Marine Invasive Species Program, which will be providing insight into assessing and reducing risks of aquatic non-Indigenous species introductions from vessels arriving in California ports.
Thank you very much, Emily. Yeah, so my talk, my name is Jonathan Thompson. Myself and Lena Ceballos-Osuna will be talking today about a little bit different subject, which is looking at more of the prevention side of things. So trying to prevent those early detections. Next slide, please. So first, I want to get into what we'll be talking about, which is managing vessel vectors from the shipping pathways. So shipping is a significant pathway for moving invasive species around the globe through the vectors of ballast water and vessel biofouling. Vessels manage their ballast water and biofouling. Vessels managing their ballast water and biofouling is the most effective way to prevent new aquatic non-Indigenous species introductions from this pathway. Next slide, please. So as vessels move throughout the world and come into California and the ports inside the Golden Gate, they have the potential to move species around with them. So shipping is responsible for up to 79% of established aquatic non-Indigenous species in North America. Next slide. So as far as the ballast water vector, how the mechanism for this vector works is that as vessels arrive at a port and they offload their cargo, as they move to another port, they'll load ballast water for stability purposes. And when they load that ballast water, they're also loading whatever organisms are in that water. So whatever port they're at in the world, they're loading whatever organisms are at that port. As the vessels move across the world with an empty cargo hold, they're taking that ballast water with them in their ballast tanks. Now, when vessels arrive at another port, they offload that, they start to load the cargo, and as they're doing that, they're offloading that ballast water, which includes the organisms that might be in that ballast water. And of course, the vessel either has a completely full hold of cargo or partially full, and then moves on to another port to do more of the business. So roughly 7,000 species are moved around the world in ballast on a daily basis. Each ballast water discharge event has the potential to release over 21 million individual organisms. Next slide, please. So for vessel biofouling, refers to organisms that are attached to a vessel's wetted surfaces. Vessel biofouling communities consist of both organisms directly attached to the vessel and mobile organisms that might be associated with those attached organisms, sort of creating a community on the vessel. These organisms can survive long voyages in a wide range of environmental conditions. Biofouling communities include fishes, barnacles, algae, mussels, worms, crabs, and other invertebrates. As vessels transit from port to port, biofouling, and this is where the mechanism for biofouling comes in, these biofouling organisms can drop off or spawn, resulting in a potential new introduction of aquatic non-indigenous species. Vessel biofouling is considered a significant vector of aquatic non-indigenous species introductions in California and other areas such as Hawaii and Australia. Next slide, please. Okay, so the risk of non-indigenous species introductions from these pathways is assessed in the Marine Invasive Species Guiding Mandate. And our mandate is to eliminate the discharge of non-indigenous species into California waters from vessels arriving at California ports. It includes any of the ports where vessels arrive. That includes Sacramento and Stockton, and the other ports within the Golden Gate. So one of the bigger ports is the Port of Oakland, but there's also ports in the Carquinez Straits. We define vessels as vessels that are 300 gross registered tons and carrying or capable of carrying ballast water. So as a result of the Marine Invasive Species Program mandate, the likelihood of an introduction through the shipping pathway is decreased by working towards eliminating the discharge of non-indigenous species. Any potential impacts from new introductions are prevented by preventing introductions in the first place. So to implement this mandate, the Marine Invasive Species Program implements requirements for vessels entering California waters. These requirements are focused on reducing the risk of introduction of non-indigenous species through ballast water discharges rather than using a species-based approach. So we're focused on managing the vector and not managing for individual species. So introductions of aquatic non-indigenous species are prevented from ballast water discharges by requiring vessels to exchange ballast water or use a treatment system. And then coming in 2022 by meeting a discharge standard. And if you would like to hear more about discharge standards, our program is delivering a webinar tomorrow on implementing these discharge standards starting in 2022, January 1st of 2022. So for biofouling, vessels are required to manage their wetted surfaces through coatings, underwater underwater cleaning, or other actions for specific parts of the ship. Coupled with outreach, reducing risk through requiring vessels to manage biofouling and meet ballast water discharge standards is the most effective way to prevent new aquatic non-indigenous species introductions through this pathway. And a little bit about how we manage a risk is, additionally manage risk is through inspections. So to ensure that the management requirements are followed, vessels are boarded for compliance inspections and to engage with vessel crews to deliver outreach. And these inspections improve prevention. Vessels are targeted for boarding based on risk profiles and available resources. For example, vessels with a history of non-compliance may be non-compliant in the future. Also, vessel crews and regulations change, so considering when the last inspection occurred is important. And we prioritize vessels that haven't been inspected in the last 5 years. So that includes vessels that are new to California. For biofouling, we look at if vessels have an extended residency period, which means, if a vessel stays in one place for longer than 90 days, and they have that opportunity for the biofouling communities to grow on the vessel. Also, we look at vessel coatings that might be outside their expected lifespan. For ballast water, vessels that are discharging have the greatest risk for moving aquatic NAS. And based on another legislative mandate that we have, our program is able to board 25% of arrivals, and that includes all of the categories that I just talked about. So non-compliance, the last inspection, and then the vector risk profiles. So for agencies or other organizations that are looking to inspect vessels with more limited resources, there's another approach, and Lena is going to cover that.
Okay. Next slide. Can you hear me now?
Yes, we can hear you.
Okay, next slide, please. Okay, so thank you, John. Now I'm gonna change a little bit the topic, and I'm gonna give you a very quick summary of a paper that we recently published, where we present a proxy-based model to assess the relative contribution of ballast water and biofouling, and how we can use that information to prioritize vessel inspections. This was a collaboration between State Lands and the Smithsonian Environmental Research Center, and it's an open access paper that you can download, scanning the code or contacting me. And next slide. Okay, so the model presented in this paper is not the current approach that California is using. Here, I want to show like a conceptual graph where we see how different approaches to assess the risk are related to the resources. So ideally, we have a lot of resources and we can go and inspect every single vessel and make direct measurements. But of course, that's not the case. So what Jonathan was talking about and what California is currently doing is a risk profiling approach. And what I'm gonna talk about now and what we presented in the paper is a proxy-based model for jurisdictions with limited resources. So this proxy-based model uses readily available data and for jurisdictions with limited resources. And what it does is that it allows data-driven decisions. And as we know, every time that we use proxy-based approaches, we have to have some assumptions. So this model uses the assumption that the likelihood of introductions increases with probable supply. Next slide. So the two proxies that we use in the model are the total wetted surface area, which is a measurement of all the area in the vessel that could potentially be colonized by biofouling organisms. And this is directly related to the size of the vessel. And the other proxy that we use, and that's the proxy for biofouling. And the other proxy that we use for ballast water is the volume of ballast water discharge. And then we calculate a score for each of these vectors relative to our own population or the vessel's population using historical data. And then when we add these two vectors, we create this potential probable pressure score per arrival. And that score is allowing the jurisdiction or the agency in charge of inspections to prioritize arrivals based on the presumed risk. Next slide. Okay, so to test the model and to show its functionality, not just for inspections prioritization, but for our allocation of resources, we use 1 year of California data. So with this model, we can add the PPP scores or the potential probable pressure scores, but by grouping them by different factors, for example, by location or by vessel type. And we also can see the contribution of each different vector. So we can see how much contribution is attributed to ballast water or to biofouling. So in this graph, in the y-axis of the bottom graph, we see all the different ports in California, and it's showing the cumulative potential probable pressure score of 1 year. And in the top graph, we divided by the different vessel types. So it just shows different patterns within our jurisdiction that will allow to see where we should allocate the resources depending on what we need. Next slide. Okay, so this was very quick, and I'm happy to talk to you about this paper, but this is just a summary of what the model is intended to do. So it's adaptable to each jurisdiction's vessels population. It can be adjusted for different segments of the vessel population. It can be used just for ballast water or just for biofouling or both. And it can be grouped by different factors, vessel type, time periods, locations. But the most important thing is that it's very simple, really easy to set up. It uses two readily available values, and it will allow each jurisdiction to have a baseline to then design their own prioritization scheme. And with that, I would like to thank you. And I don't know if we have time for questions.
Thank you, Lena and Jonathan. We don't have time for questions, but we are about to head into our panel discussion. So if anybody has questions for Lena or Jonathan or any of the other speakers, please put them in the Q&A. And so thank you again, everyone, for your excellent presentations. And the panel discussion up next will go until approximately 12:30. That will include our presenters as well as Valerie Cook, the program manager for the California Department of Fish and Wildlife's Nutria Eradication Program. And the panel will be moderated by Luis Conrad, Deputy Executive Officer for Science with the Delta Stewardship Council. And yeah, we will go into the panel.
Good morning, everyone, and thank you, Emily. Thank you for all of the speakers that we just heard from in their presentations. I'm going to briefly run through your names to remind the whole audience of who we have. We have Jared Barr from CDFW, and joining him, although didn't present in the session, is Valerie Cook, also from CDFW. And we have Jeff Caudill from State Parks Division of Boating and Waterways, Ron Vanderhoff from the California Native Plant Society, Jonathan Thompson and Lena Caballos-Asuna from the California State Lands Commission. If all the panelists, if you're able to, I appreciate your turning on your cameras for the question and answer session here. And I'm going to start off with a question that I'd like each of you to spend just a minute responding to. And after that, we will prioritize any questions that have come in through the Q&A. And if we don't have questions, we'll continue the discussion until 12:30. So that first question is, what challenges and barriers to implementation of early detection and rapid response are most important, do you think, to address in California? And what about the Delta specifically? And Jared, we'll start with you from CDFW.
All right, thank you. So yeah, regarding the Delta specifically, at least in terms of Nutria, the Delta is a large geographic area, and habitat in the Delta is very dynamic. With tidal influence and with shrinking and expanding habitat for Nutria, I think that presents a large challenge for us in terms of monitoring and getting the resources we need to be able to detect animals where they're present and then respond appropriately to that. And that's what I can think of off the top of my head right now regarding the Delta specifically. Valerie, do you have potentially anything you want to add on to that?
Yeah, I think Jared really highlighted a lot of the issues that we're facing in the Delta in an area where we really need to be able to work most efficiently. But the reason I joined this group for the panel was because Jared and I kind of have different perspectives in the program, where I was involved from the initial ICS stages and Jared's involved now. So I can speak to, when the initial detection of Nutria occurred back in 2017, we faced all of the challenges right up front, where I don't think any agencies in California are sitting on like a rainy day fund to facilitate rapid response when an incidental detection occurs. So, we knew that Nutria was a huge issue. Somebody had to respond, but, nobody was sitting on staffing, funding, resources, equipment, fleshed out, authorities, access. We really had to start from scratch, and that's where the ICS initial framework kind of came into play. But I would say if we had to point to one major challenge, it was securing adequate funding and resources to be able to implement any kind of a response at all.
Thank you, Valerie. That gets to the really heart of the issue, I think, is you've had to be able to mobilize quickly. Are any of the other panelists want to jump in next on the key challenges to implementation?
Sure, I can speak real quick. One of the challenges that our program has is we're geographically limited. So if a source population of an invasive is outside of the Delta, a control program is perhaps the best that we could be, unfortunately, because we're not able to control that source. I agree with both Valerie and Jared that collaborating with others is important because the monitoring is critical. In our case, if we had known about alligatorweed even a year before, if it was there at that time, we're not sure. We might have been more successful in controlling and potentially eradicating alligatorweed. And same thing on the resource function. If a new set of invasives comes through and we are not properly resourced to be able to handle that particular challenge that may come in, be it funding or just personnel, it could take us years to get to that point. And I'm not entirely sure that a 3-year response would be a model for EDRR.
Thank you, Jeff. And Ron,
I can speak.
Weigh in?
Yeah, from my area and plant specific, I would say my challenge is data sharing, and sometimes the challenges of that data sharing, particularly because we're focused on the newest detections. Our objective is to deal with these so that they never make it to most of the people in this audience and they're no longer management concerns, and to get them right at the very, very beginning. In order to do that, we really need to understand where these things are, or where, and where they're not. And it's interesting that in so many occasions we've made a detection, and then after reporting it, we find out that, oh yeah, well, we've had that for 3 years, and we just, have it in our little database, and the guy across the street, across the fence, didn't know about it, and we're training volunteers and so on. So anyway, agreement on data sharing and what that data platform is. And I know there's lots of obstacles to that, and we just need to work through those.
Thank you, Ron. I do want to dive in on early detection in a few moments, but first, Jonathan and Lena, would you like to talk about major challenges that you see with your program?
Well, our program is mainly focused on prevention, but as far as early detection and rapid response, I think one of the biggest challenges, and I've heard some of the other panelists say this, is that just a dedicated control fund, so funding for, a rapid response fund is what I meant. So that dedicated fund for that, and then also having, at least for the Delta and for some of the marine areas, is who's going to respond to new introductions if they occur.
Lena, anything to add?
No, I think that's it. All the challenges that we face are mostly on how to prevent and how to properly assess. Let's say right now we're working on assessing compliance for the standards, and it's very challenging to actually look if vessels are compliant and if they're not bringing anything. But yeah, we currently don't have early detect response role, let's say, in the same way.
Well, thank you. Thank you for that reflection. So I want to ask Aaron, do we have any questions coming in the Q&A that we should address right now?
We have one from earlier in the symposium that would probably be interesting to have the panelists weigh in on, but nothing new and fresh.
Okay. Maybe what I'll do is dive in, and if we get to a pause, then let's address that question from earlier in the symposium. Thank you, Aaron. So I wanted to ask the panelists a little bit more about this early detection piece, which is so critical to enabling ourselves to be able to have a rapid response. And Jeff, your reflection that it would have been great to have an even earlier detection of alligatorweed was really helpful. And wanted to ask the panelists, what do you think we need to enhance our ability to have early detections? And I'm wondering if you can reflect specifically on whether we are leveraging a role of citizen science to the extent we can be. And Ron had some great reflections on what it takes to engage volunteers and the community and as eyes on the ground. Many state agencies, I think we can all reflect that we would love to have more resources and more boots and eyes on the ground to detect new invaders, but we are limited with our staff resources. So how do we engage others? And then a second piece to this early detection that I also want to see if you have any thoughts on is that I think Lena gave an excellent overview of a model-based tool that helps us to prioritize where to look. And to what extent can we be employing modeling tools, or have any of the programs been thinking about what additional quantitative tools could we be using to provide a framework for our own early detection abilities. So citizen science and modeling, any thoughts on how we can enhance our early detection ability? And I'll let anyone jump in if no one's volunteering, I'll try to ping you.
Well, I can start again from the South County or South State. Being mostly engaged in volunteers and early detectors, there's not a central method in which to report a detection. And it's oftentimes that's a very mysterious challenge to people who are not, professionally engaged in this. They don't know what to do. They think they saw a new something that, they're maybe pretty good at their plant IDs, but there's no real, what do I do with this information? And I think a lot of times it just goes nowhere. It just sort of stops at that point. And it would be great if there was a platform for that. And ideally, it would not be a land management organization because they have boundaries. It would be, I'm going to make a plug for Doug at Calypsy. It would be someplace like that, which doesn't have any boundaries, and everybody would participate and acknowledge that's the place for early detection reporting by the citizen science world and the field botanists and the people who aren't professionally out doing surveys. The other thought on that would be to engage platforms like iNaturalist and CalFlora to a much greater degree. Those are where hundreds of thousands of observations are being made. And I can't tell you how many times I've monitored that and I find a new detection in Orange County that somebody just posted out of the blue. So, acknowledging those platforms and engaging those platforms and gleaning those platforms, and then having a central repository, if it's not one of those two, that we can all forward detections to.
Thanks a lot. It's a great acknowledgement that there already are many eyes and on the ground, and they are recording data. So any of the other panelists want to weigh in on this?
Yeah, this is Valerie. I'll go ahead and chime in with a few efforts that we're aware of. So prior to the Nutria detection in California, there was some modeling done, and there were some publications that modeled risk of establishment throughout North America for Nutria should they be introduced. And so that was, kind of a foundational paper. The department's interagency ecological program has also been working on some modeling efforts to look at locations where Nutria have been detected or found, to see if they can extrapolate and come up with any useful guidance on, probability of dispersal or establishment to help us target our efforts. But frankly, our reality is, what we're observing is if there's aquatic habitat and it's, not washing them away, then it's pretty well suitable if there's some vegetation there. So another piece that we're also looking at, which DWR is actually funding, a population genetics analysis. So we're looking at kind of the distribution of genetics across the landscape, not only to be able to determine what proportion of the population we're taking as a proxy for, what else is out there that we might be missing, but also looking at modeling the distribution of genetics across the landscape to help us help elucidate dispersal trends in males and females and juveniles and family members, and to help us understand dispersal distances as a way that we can potentially model kind of risk of spread from known populations to help us evaluate to make sure that we've got adequate coverage on the landscape. So lots of moving pieces that are coming together to help us make some informed decisions moving forward.
Thank you, Valerie. It's good to hear about some modeling tools coming to bear on the Nutria issue. Jared, do you have anything to add on that?
I was just actually thinking about the first part to the question.
Yeah.
So improving early detection. And, you know, for our program, I think what has helped us is continuous community involvement. We are constantly sending out mailers to communities, and we have Nutria pamphlets and lots of outreach materials, and try to educate people as much as we can so that they can identify Nutria, and kind of be our eyes and ears a little bit as well. And we always encourage folks to reach out to our general email, and whatnot. So I think having a kind of shotgun approach to having our hands in a lot of different places has helped us out in detecting individuals where, you know, our field teams can't be at all times. And yeah, I think that's a big piece as well. We have, and I mean, there are caveats to that. We do get, typically when we send out mail or outreach materials, we get a lot of pictures in our email of feral cats and raccoons and all kinds of stuff. So, you know, sifting through and trying to make sense of reliable observations is part of it too.
Thank you. So Valerie, David, I want to say I see your hand, and I'll want to make sure that if Valerie was on this thread, do you want to respond or build directly on that? Because your hand's up, Valerie.
Yeah, I think Jared just touched on it really. I think partnerships is really another piece of that as well. Jared mentioned a lot of our outreach efforts, but the Nutria program, we rely on so many partnerships and so many informed people that also help us spread the word and are giving presentations and conducting outreach to their stakeholder groups. So David Kratville, Gina Darren, Jared, Calypsy, I mean, across the board, same even with boating and waterways. I mean, we have very strong relationships and partnerships with these other agencies that really help us get the message out there and spread the word about how to report either directly to us, or our partners are relaying that information to us as well. So I think partnerships come into play there as well for us.
Thank you. And David, do you want to weigh in here? I can't hear you, David. You can try one more time. Okay, no, I can't hear you. Okay. Perhaps you could put your question in the Q&A or somewhere and we'll try to get to it. Okay, thank you. Okay, so we're talking about early detection, and Jeff, just want to acknowledge I know that this citizen science may be an area that Boating and Waterways does think about, work on, and just thought you might be interested in highlighting what your programs work in that area, maybe.
Sure. As others have said, the resource limitations make it so citizen science is almost a requirement in order for any kind of early detection or collaborations that we've got, and that goes, you know, beyond working with other partner agencies and such. I mean, so we're working on an app that we hope to launch at some point here soon, which would allow folks to make reports regardless of where they're from, as long as they have the app to you know, let us know in the Delta what they see, attach a picture, give us coordinates, or just kind of describe where they're at. And this could help with having folks identify new plants as well, which would help with, say, the new invaders, because we don't have enough, we'll call it boots on the ground, to be able to cover the entire Delta to look for something like this.
Sure.
And so if we're not able to hear from others about an impending invasive that may be going into the Delta, that may also be an issue of concern that will outcompete other natives and invasives within the Delta, this is another option that we'd like to utilize. It also lets folks communicate with us and let them know, or let us know also how we're doing on top of it. But there's a lot of folks in the Delta that really care about the Delta, and they spend more time on the water than we do. So, you know, giving them the ability to contact us very easily would be very beneficial. And so that's something that we are looking into and hopefully being able to, utilize sometime this year or so.
Thank you. We look forward to that. That is exciting to hear about a new tool potentially coming out. I just did take a look at the Q&A and I see that there are a few questions there. We just have 1 minute left though. So I am going to, let's see, Jeff, I think that one of these about ribbonweed is an invasive plant new to the Delta and also to North America. I just want to call your attention to that. Who is responsible for the EDRR in this case? And maybe you could type an answer to that in the chat. With that, I wanted to give the panelists a moment, if anyone would like to weigh into one other broad question that we have, which is, what are the challenges you see to maintaining your program over time? These are programs that need to be continuous and always watchful. Are there funding, program partnerships, other aspects that you think really want to highlight the need to continue in order to have continuity?
I can start really quickly. Sorry, Valerie, for not raising my hand. Yeah, I think we're currently resourced as a control program. And so, for example, as Ron had mentioned earlier, data sharing is very important, because without that data sharing, it's almost like we're all kind of reinventing the wheel. And so higher level of collaborations, data sharing, utilizing other opportunities such as citizen science, I think is critical to being able to utilize EDRR. There's also the issue of resource constraints as well, which might be a much tougher ask for all of us involved. But I think starting with the early detection gives us a lot more options, because that's an opportunity for us to utilize all the subject matter experts around the table here and who operate in and around the Delta to be able to figure out what is our best course of action given our limited resources and mission.
Thank you. Any final thoughts from any of the panelists on challenges to implementation and keeping the program alive? And Valerie, you have your hand up.
Yeah, I think I would touch on two topics. So both funding and staffing. So with funding, whether it's a control operation that's ongoing and indefinite, or an eradication effort, which is inherently long-term just due to the nature of the need for those final several years of monitoring to make sure the population is gone, you have to have stable funding to continue those operations, even for those years where you're not getting any detections, and it's, can be looked at either you're ineffective or you've done your job. But so I would say stable funding, because, often folks have to go to grant funding opportunities to support operations, especially early on. And those are short-term funding solutions. So for an eradication effort, we are still in the pursuit of long-term stable funding to see this through to the end. But the other challenge that comes with that, these granting opportunities are fantastic. They've carried us far. We've fared well with them so far. But the reality is, when you have a short-term funding source, that doesn't come with permanent positions. If you work for, the state, you don't have permanent positions. You can fill those as seasonal or limited term positions. So every, 12 to 24 months, you have to let a person go and hire somebody brand new and retrain them. And you have this constant turnover where you're losing your expertise. And so as you need, as the program progresses and you need to rely on that expertise and that experience, and you're constantly losing it, that is a real challenge for an effective and continuous program. And I will say that luckily we were able to successfully, we had a successful budget change proposal this past year. So we were able to transition all of our limited term positions over to permanent so that we can retain that expertise and that skill set that we've invested in. But that's a real challenge too that comes along. It's not just a matter of money, it's the ability to bring in good people, train them, and then retain them so that you've got your most effective people operating on the ground.
Thank you. That is consistent also with David Cradfield's reflection on the institutional knowledge and how it doesn't it's a resource. And Jared, do you have anything to add? Your hand is up.
Yeah, do we have time for another?
We can, well, just a brief, I want to make sure you could, we'll close after your comment.
Okay. Yeah, I just had one additional thought, at least relative to our program, which might resonate with other folks. We rely heavily on partnerships with public and private landowners. And that's something that we really need to maintain throughout the life of our project or program in order for us to actually get out and monitor and survey. If we don't maintain those partnerships, we essentially lock ourselves out from areas where our particular invasive species might be occurring. So that's something as well that we find very important in our program.
Thank you for that. And also, I think that's probably linked to the institutional knowledge, because those partnerships are when it comes down to it, often with people, not with entities. So when there are people that have those relationships and partnerships, they can be leveraged. And when those people go away, you lose that. So thank you for all of that. Thank you for all the talks. Thank you for the conversation afterwards. And I will close the Q&A and the panel session now. Turn it over to Rachel so she can guide us through next steps before we break for lunch. Thank you.
Excellent. Thank you so much, Louise. And thank you to all the speakers and the panelists who have talked already today. I feel like it's been really exciting to learn from all of you all. We are going to break for lunch now, and we will return at 1 o'clock. So we ran a little bit over, but we'll try to get started again right at 1. So if any of the speakers or organizers that are in the afternoon sessions want to stick on the call for just a second so we can coordinate, but everyone else, feel free to break for lunch, and we look forward to seeing you back at 1 p.m.
Next up for our talk in the session is about range shifts. And so next up we have Jorge Renteria, from a postdoc fellow at University of California, Davis. So Eric, if you can please pull up those slides and then go ahead, Jorge.
Hi, Rachel. Thanks, everyone. And I'm Jorge Renteria. I'm here to present I'm going to talk about assessing the risk of plant species invasion under different climatic conditions in California. This project was developed as a part of my postdoc at UC Davis, working in collaboration with the California Department of Water Resources. Next slide, please. Before to go into the specific project, I would like to provide a background of how this project falls within the EDRR response and climate change. Most of the stuff has been discussed with the previous presentations, and so I'm not going to get in deep, but the important thing is that when different measures to stop invasive species fail and are not able to stop the arrival of potential invasive species, an EDRR system might help to identify, contain, and perhaps prevent future problems with a potential invasive species. An EDRR system normally requires a series of information about the species distribution, assessing what is the risk, which institution will be the ones that they are in charge of the management, and also defining the best methods to control and implementing the actions plans. Next slide, please. I have summarized here an EDRR process, and or a specific project I'm going to present falls within the risk assessment. And as it was present in the previous presentation mentioned, there are several risk assessments, and each one differ and can be, sometimes they can be complex or time consuming depending on the requirements. And normally they rely or they are focused on questions about the specific species, and even there are several options to go. What I found in my experience that the main constraint is that there are often no enough information to answer all the questions that the system requires. Next slide. So in terms of climate change and invasive species, as like the prior presenter mentioned, there is an expected impact in all the organisms. And in a specific, it's expected that climate change or the future climate conditions are going to affect species in different ways. And some climate conditions are going to be favorable for some species, and the same conditions will be adverse for other species. And that applies for native and non-native species. In the context of invasive species, climate change is expected to promote the establishment of additional species. Also, it's expected that some non-native species that at the moment don't seem to be invasive might become invasive because favorable conditions are going, because there are going to be favorable conditions that allow the species to become invasive. And also the impacts are expected to change, as well as is expected some change of the distribution of the species that are already invasive in one particular place. In summary, with climate change, it's expected that there are going to be winners and losers. Next slide, please. So I'm going to take this opportunity to present this publication, which came out early this year. And basically what we're trying to do is trying to assess the risk of invasion of a new ornamental plant species in California, and using as a predict factors future climatic conditions. This assessment uses a relatively simple methodology and provides a basic and rationale to identify and prioritize areas and species that can be of concern. The evaluation also try to identify hotspots of invasion, and also we run an individual risk assessment for each of the species that they was considered in the study. Next slide, please. So in terms of the methodology, we take as baseline a list of species or plant species that was produced by Brusati and et al. In 2014. And this list of species is a list of ornamental species, and the assessment was based on the knowledge of these species being invasives elsewhere, and species that they were coming from Mediterranean climatic conditions, or those species were known to be invasive in the neighbors states. We took this list, and they come up with a list of 180 species that with a potential high risk of invasion in California. So our approach, we took this list of species, and we tried to refine the species, assuming that if this species represent a risk, there'd be a way to rank the species, because our rationale is that the risk of the species were not similar across all the species. So the main tool that we use for this analysis is a species distribution modeling. And species distribution modeling use the combination of species occurrence with some climatic variables. So in order to run the model, we need a minimum of occurrence, species occurrence. Therefore, of the 180 species that they were listed by Brusati, we only work with 170 species for which we were able to gather enough records of occurrences. And once the models, we create the models, we project the models into California current and future climatic conditions, and we create this prediction for each individual species. And in order to create the risk map, we combine all the prediction of every individual species, and in that way we were able to create a hotspot of invasion. As part of this project, we also run an individual risk assessment. And in order to do that, we combine the predict area that was generated by the species distribution model, and we, in combination with some species biological traits that are novel to facilitate the invasion process. Next step. So as I mentioned, we use our main tool that use for this study was species distribution modeling. And in generally what they do, the models they do, or species distribution models, they are basically use a species occurrence in combination with environmental variables, and through a statistical analysis, try to describe the best conditions where a species can be established. Once the models has been run and fitted, I mean, there is a process of evaluation, and when there is a certain confidence that the model prediction, is possible to use these models to predict the potential distribution of a new species in a new area. For this particular study, we use the species occurrence of the 107 species, the occurrence that come from the native and introduced range. And we use that in order to describe the full extent where each particular species can establish. There are a series of different algorithms or models that they can be used, and each model can produce different outputs. And so we use 3 different models in order to reduce the variation of each model. And to project the distribution, we use 2 different climatic scenarios, which are called GCM. Basically, these models provide information about, or estimation of what will be the climatic conditions in the future. It's not possible to see in a slide. I'm going to describe a little bit more later. Next slide, please. So the individual risk assessment. So in order to run the individual risk assessment, we use 2 parameters. One is related about the potential distribution that was generated by the modeling approach. And the other element was the species biological traits. Those traits are known to be related with, or that are known to facilitate the invasion process. So we score each of the parameters in a score system that goes from 1 to 3. So in terms of the predicted area, we use the gross predicted area for each species, and obviously a species with a wider distribution have a higher score. Also, we combine this distribution and see how many ecoregions overlap with the potential distribution, and a higher number of ecoregions means a higher score. In terms of the future climatic conditions, we use 2 different climatic scenarios. So in the case that the species only increase the area in one scenario, we provide a low score, the 1, and then a higher score if the species was predicted to increase under both climatic scenarios. For each GCM, we also use the prediction under 2 different greenhouse scenarios, but I'm going to explain later. Next slide. Here we have the results, and in the left we have the potential distribution of the mass risk that we generate using, on the left side, the one on the left is under current climatic conditions. And what it shows is the species richness that are predicted to occur in California. So obviously the red or more, the close to red, it means that more species have been predicted in each cell. On the other 2 graphs on the right, we have the 2 GCMs, GCM CNRM and GCM MIROC. Those 2 climate scenarios are two of the GCNs that having a kind of have simulate and accepted, like simulate in a good way the climate conditions in California, and they represent, each one represent extreme conditions. And so here we can see that the patterns are similar across the two models, and we don't see any major difference. So, however, in the table we try to summarize what was the change of the risk, and a positive value means that the category risk has increased, and a negative value means that it has decreased. And we will see that under the mirror scenario, and we see that the high category risk species tend to increase. Next slide, please. So here we can see an individual level that the species that a number of species, the species that are under influence by the mirror scenario tend to increase more in area than those ones that they are the area that is predicted on the GCM CNN. Next. Here is we present the risk, the individualized species risk assessment, and even it's a long table, but in this slide we are just presented the top 10 categories. And the important thing is that we were able to rank the species using the two elements, the potential distribution and the biological traits, in order to rank the species. And this risk system, or this categorical system, can help us to identify or prioritize species at different levels. Next slide. Well, in summary, we can say we know that preventing, controlling invasive species is a high priority. However, often is the case that we have to make decisions without time. And we provide a relatively easy methodology, and we were able to identify hotspots of potential invasive species. And this is the first stage, and more information can improve the prediction. The risk map can use a different regional levels in order to survey particular areas of the species. And also, depending of the status of the plant and the risk, and different measures can be taken for each particular species. Thank you.
Great, thank you, Jorge. There's no time for questions. We're going to roll on to the next presentation, but there is at least one that's in the Q&A. So hopefully you can take a look and add your response there. So next up is early detection monitoring talk for invasive mussels in California. So Tomas Javush, Senior Environmental Scientist Supervisor in the California Department of Fish and Wildlife's Invasive Species Program. So take it away, Tomas.
Okay, thanks for the introduction, Rachel, and good to see you. And good afternoon, everybody. So yeah, I'm gonna talk about early detection monitoring of invasive mussels in California. And since I'm leading the state's, the California Department of Fish and Wildlife's Aquatic and Zebra Mussel Program, this talk is about those two guys, and we heard about those before in other talks. And there's basically three things I want to highlight in my short 12 minutes. One is reiterating our dire need to reprioritize our resources in light of, this look for the needle in the haystack. So, and put some perspective on this. So we got about 3,000 lake type water bodies in California with names, and that's only those, not even including any delta waters or rivers, etc. And we got about just over a dozen staff that are actively doing fieldwork in the program across the state. Okay, so that's my second slide. So the third point I wanted to make is that actually Second point was I'm going to introduce something that we developed to assess, to look at habitat suitability, basically Stephen Brand's ingredient number 2, or call it risk of establishment, as a guideline to focus our resources and effort. We do, of course, consider risk of introduction and impacts as well that mussels have, but we don't have a formalized process for that, but I'm going to talk about this a little more in a moment too. And third, I wanted to also highlight our continuing data collection effort. We are also collecting environmental data to continuously refine our early detection monitoring. So, and now to this slide that came up a little early. So, not trying to beat a dead horse. So, yeah, resilient mussels though would be really bad news for the Delta, given all their potentially adverse impacts on ecosystems and connected to that fisheries, but also, you know, Delta as a place to recreate, boating, other recreational opportunities, as well as water delivery. They've been known to cause damages in the billions in other systems to water infrastructure and delivery. And you know, considering how interconnected the Delta is with everything else in California in terms of water. So next slide, please. Okay, so quagga and zebra mussels haven't been found in the Delta, so that's good. The closest invasion to the Delta is in the Central Coast area, actually Central California, in San Benito County. San Justo Reservoir mussels were detected there in 2007. And that's kind of also when our early detection monitoring program started. And all the other the invasions are actually by quagga mussels are in our further south in the state. And quagga mussels were found also in 2007, 2008. You saw this a little bit already in Teresa Tong's talk. They came from the Colorado River, and all the initial detections were in reservoirs that received water from the Colorado River. So there were about 20 detections in those initial years, 2007, 2008, and the detection rate in additional water bodies has been considerably slowing since then, due to prevention and control efforts kicking in. So, and I should probably also mention that most of the additional detections, the introduction of mussels was most likely due to overland transport with trailer boats. So next slide, please. To tell you a little bit about our program, so CDFW's statewide quagga and zebra mussel early detection monitoring program is carried out both by state headquarters and the CDFW regions. You'll see a map of our regions here, specifically the inland regions, regions 1 through 6. And state headquarters is the lead mostly on the planning and coordination, and then the regions are implementing the monitoring. So for the Delta, Region 3 is the region in charge, the Bay Delta region, and that's because most of legal Delta is located inside Region 3. There's others in the state monitoring for quagga and zebra mussels. For example, the California Department of Water Resources and the US Bureau of Reclamation. Both these agencies also are monitoring the Delta. And then there's also many regional and local water agencies that ranges from big players such as Metropolitan Water District to small agencies, and we either compel or encourage them to conduct their own monitoring. So next slide, please. We're also offering resources for others monitoring quagga and zebra mussels. For once, we're offering training and sampling. We've developed standardized monitoring protocols that we got posted on our website for everybody to use. We got a lab in Bodega Bay where we conduct analysis of plankton tow samples that are submitted or species identification. And we also developed the monitoring recommendations that I'm going to talk about. So next slide, please. But before I go there, just briefly about our monitoring methods. So there's methods that are good for detecting actual mussels, juvenile and adult, either by surface surveys or artificial substrate. Surface surveys is just checking walls and rocks and docks and boats, etc., for the presence of mussels at a water body. Absent any surfaces that can be easily surveyed, there's also the option to use artificial substrates. That can be something like the 100SD device shown here. That's a little more sciencey. Can also be something as simple as just dropping a PVC pipe in the water and checking it periodically. So both these methods are pretty low tech and low cost. And then we heard previously already about plankton tows. That's looking for the larvae or veligers in the water column. That's a little more involved, towing nets around the water and collecting plankton in an attached bottle, then processing it, preserving it, and sending it to the lab for microscopical analysis. So next slide, please. Okay, so this is a graphic representation of our recommendations. This is available on our website. I don't want to get into many of the details, but basically I mentioned this based on habitat suitability. It was developed based on a very extensive review of the mussel literature and their dependency on environmental parameters. So the four main parameters that we're looking at are calcium. That's the single most one actually. Calcium, pH, salinity, and temperature. So in this graphic is mostly based on calcium, and we're dividing water bodies into categories based on calcium and have some thresholds, different categories ranging from very low to high calcium. So, and then there's a breaking point here that you see. And I just want to give two examples how this is being used to, inform the intensity and the methods used at a specific water body. So moderate and high calcium water bodies are water bodies where mussels are capable of completing a life cycle where you would actually expect to potentially find veligers, or could expect to find veligers if they were there. So that's where I recommend plankton tows. And then we would also increase the frequency of plankton tows depending on the temperature. So if the temperature is in a range where conditions for reproductions are optimal, we would increase the frequency. On the other hand, in water bodies that are in the low or very low calcium range, we would recommend don't bother with those plankton tows because we wouldn't expect veligers to be found to begin with. Just do some surface surveys to do your due diligence. So those are just some examples. Of course, we take these other parameters into considerations. We don't go sampling in water bodies that are acidic, that with a pH less than 7, and that are too salty for mussels, that have a higher salinity than 6 parts per thousand. So to give you an example, next slide, please. This is very oversimplified graphic of the delta and how we would monitor in different regions. So not taking into consideration site-specific things, like, for example, on the west side of the Sacramento Valley, you often find high calcium sites due to the coastal range influence. But the general picture is Sacramento River water is very low in calcium. So most sites that are dominated by Sacramento River water, you would expect low calcium conditions. We would say don't bother with plankton tows. Moderate to high calcium conditions are in the South Delta to be expected because of the San Joaquin River influence. The San Joaquin River carries high calcium water. And if you get outside of the confluence, salinity gets soon too high, and there's some we would probably not monitor for quagga and zebra mussels. Now, yeah, this is very oversimplified. As I said, the delta is a very complicated system, lots of temporal and spatial variability for water quality. Next slide, please. The third aspect, our environmental data compilation and use of it. So when the program started, one of the first things was looking at the potential distribution of mussels in the state, based on these parameters that I mentioned, and mostly calcium. So here is a map that includes the delta. We had data for about 160 water bodies at the time, or sites, let's say sites. So white circles are sites that are not suitable for mussels, and black circles are sites that are for mussels. And yeah, it's just to make this point. So you see the density of data points here. There was a lot of extrapolation needed for water bodies where we didn't have data. But meanwhile, our compilation efforts, especially in recent years, have intensified, and we also got grant funding to collect additional water quality data. So next slide, please. So our database is growing, or filling in data gaps, and so our information basis is much denser. So, and yeah, you can see that if you compare the two slides. These are sites that we have data for at this point. Next slide, please. So, and then again, there's of course other considerations for prioritizing monitoring efforts. And as I said, we don't have a formalized process for that. It's basically us getting together with the regions and discussing these other aspects of risk of introduction and potential impacts on a case-by-case basis, like is there public access, how much boating or other recreational activity, how close water bodies are to infested water bodies, or whether they are hydrologically connected. Those are all aspects of risk of introduction. Then, of course, potential impacts, for example, what's the ecological significance of a water body for example, for threatening endangered species, or potential economic impact, importance as a water resource. And of course, last but not least, also coordination, who else is monitoring how and how frequent. Next slide, please, which will be my last. So take homes again, there's an obvious need to prioritize and focus early detection monitoring efforts. We developed science-based monitoring recommendations for us and others to use. And we're also making use of available environmental data and filling data gaps, which is helping us to refine our early detection monitoring planning. Last slide. So thank you, everybody, and glad to take any questions you have.
Thanks, Tomas. I'm not seeing any questions that have been typed into the Q&A yet, but I do invite folks to add comments there and questions. But thank you for that run through of everything. It's exciting. We are going to jump now, just for time to keep us on track. We're going to jump to the next talk. So next up, thank you. There you are, Melinda. This talk will be focusing on using genetic approaches to detecting species. So Melinda Bearwald, Environmental Program Manager with the California Department of Water Resources. Please take it away.
Great, thanks, Rachel. Okay, next slide. So I wanted to start off this talk by giving an overview of what eDNA is, so that we all get on the same page with that, as well as the benefits and the limitations of the methods, as well as talk about a little bit the challenges that come with using eDNA in a management context, as well as the recommendations that I'd put forth for incorporating eDNA into management decision-making in terms of best practices for eDNA use. And then finish it up with what's to come in terms of new technologies that are here or on the horizon for eDNA use. Next slide. So to start us off, eDNA really has, a fairly simple definition, which is basically just DNA being released by an organism into the environment. And that DNA can be collected from really any substrate within the environment from which you think that the individual may have been in a particular region. So for instance, in the Delta, if you want to look at aquatic organisms, you might want to sample the water to collect the DNA from. And once you've collected the substrate, for instance, the water, in the field, then you can take it back and into the laboratory and do different genetic approaches depending upon what your specific questions are. So for instance, if you want to look at just one or a few species to look at their species presence, one thing that you might do is use a PCR-based approach to do that. Alternatively, if you're more interested in getting a more holistic community-wide perspective of what's happening in that particular environment, you may instead choose to do high throughput sequencing, for instance, metabarcoding, to look at many species simultaneously for a biodiversity assessment. Regardless of which genetic approach you use, one of the cool things is that pretty much that same bottle of water, using our Delta example, would be your source for identification. So that same bottle can give you both of these approaches and answers. Next. So eDNA has several advantages that really make it very amenable for the use of invasive species detection. One is that it is considerably less labor intensive to be able to collect the DNA from the environment than to find an individual from the environment. And that's particularly true when we're talking about species of very low abundance or that are cryptic in nature, secretive, things like that. And so another area here that you can see on the lower right-hand side is the idea of what is the most effective, cost-effective method that you might want to employ. And what's been found is typically that when a population has a low density and the sampling requirements to find it would be high, eDNA can often be the more cost-effective strategy to use. And additionally, it is considerably more sensitive than a lot of different detection approaches. And so therefore it can be particularly helpful for using this approach when you are looking for these low abundant species and can allow you to detect them, for instance, in the case of invasive species, considerably earlier in the initial invasion process than what you would find for a different monitoring technique. It also has the ability to taxonomically identify species with tremendous accuracy. So you don't have to worry if a species is taxonomically very similar to another species, you can genetically tell it apart. Next slide. So despite all of these great advantages to genetics and why eDNA is such an up and coming area for invasive species, there are some things that are very important to keep in mind when considering using it. And so one of them is that really for detection of when you want to get hard counts on a species, knowing exactly how many there are in the environment, that is something that is still quite, the results are mixed in terms of what eDNA can do for you there. Some studies have found very high correlation in the eDNA counts, either the read counts for sequencing or in the amplification curves for PCR. So very high correlations with abundance, while others have not found that same correlation. So quite variable results as of yet. It would have to be very study specific for you to have confidence in any abundance estimates. Additionally, life stage is another area where really at this point in time, it's difficult to know when you just have a piece of DNA, what life stage that could possibly have come from. And so if you need to know that, again, this is a method that, might not be the most applicable. However, for the life stage, there's interesting new areas such as the use of DNA methylation to serve as epigenetic clocks, as well as eRNA, environmental RNA, to serve as a way of looking at different life stages and allowing you to assess that. And so this actually is a field that's moving very rapidly. on top of that, DNA presence, this is a very important point. DNA presence does not mean necessarily that the organism is present. And this really is something that, you know, from a molecular standpoint, we've known for a long time, but I think the communication on this hasn't always come through. And so there's different things that could cause DNA to be present in an environment and the organism not be there, such as that the organism was just passing through the area, or that it has subsequently died since, and that its DNA is persisting, or another organism could have brought that organism's DNA into the system. Either humans do that through, for instance, boat trafficking, or other species bring their gut contents in, or other things. And so it's something that I wanted to point out here, is that this is an example of something that is not actually what we would term a false positive in the true sense. And that is because in reality, what this is doing is exactly what we asked it to do. Genetics looks for the presence of DNA. The DNA actually was there in the environment, and so it did find it. What instead we need to do a better job of is communicating how that DNA could have been in that environmental place, and understanding, our risk tolerance for knowing, is the species there or not in relation to that. Other areas to consider for limitations are sample contamination in the field or lab, DNA degradation rates that can be variable based on the environment. So in the upper right-hand side, you can see different environmental variables that can affect DNA degradation rates. And really it could be temperature, or a host of different things can really cause us to have DNA degradation happen over time. And then on top of that, different organisms do shed DNA at different rates. And so basically what that means is some organisms really are shedding abundant amounts of DNA, while others are not. And that can make some a little bit more amenable to eDNA in general. On the lower right-hand side, you can see there a variety of different things, however, that we can do to mitigate both the false positives and false negatives. So the field's really moving quickly in terms of trying to ensure that we get the best results possible. Next slide. So for the challenges for management use, there are several things that, it's really something that geneticists are trying to think of doing a better job at communicating with managers. And this needs to be something, I think, that, if anything, it needs to be at the forefront of any eDNA project, the communication process between geneticists and the managers. They're, in general, because the field is relatively new, not that new. Everybody says new technology, but it's been around for over a decade now. And so there still is a lot of new development in it. Genetics is rapidly evolving. And so there's a lack of standardized field and lab protocols because people are trying new things. But there are best practices, absolutely, and we need to adhere to those. And we additionally, there are new standardizations for what should be done for any eDNA detection projects that you're going to be starting out with. Also, there is a lack of a framework for evaluating the costs of a false positive or false negative in relation to your risk tolerance for having to go out and do additional monitoring, and what a species invasion might mean for you. And lastly, there is uncertainty, as I mentioned in that last slide, about what a false positive detection might mean. But ultimately, that also comes back to the idea of increasing that communication between the geneticists and managers, so that we all have an equal understanding of that process. Next slide. So with all of that said, there really has been this paper recently came out, and I really love it and encourage folks to read it if they're at all interested in the field of eDNA and how to incorporate it into management decisions, because it lays a framework for what are the essential things that need to be done prior to starting out with an eDNA project. And I think it's right on in terms of what it recommends going forward. So for instance, getting those standards and best practices in place from, beforehand, making sure that you have validated methods. Additionally, having that operations and communications plan and talking with the resource managers is essential. So what is the criteria for saying that there is an eDNA positive in an area? And then the risk tolerance, again, what is your risk tolerance? Is it high, is it low? And evaluating that alongside the managers. And chain of custody for samples in case, legal action is required, as well as once, you have this underlying framework. What I would hate it if you got a positive eDNA detection and that's when you first ask the question, okay, so now that we have this, what's the next step? You should already know what the next step's going to be if you get an eDNA detection, really before you ever begin this process. And so that over here on the right-hand side shows you that this is just an example of what that could look like in terms of a flow diagram. So a decision support tree, really, of what during an eDNA sampling process you could consider. So it's important to keep in mind when you're doing eDNA sampling that a single positive detection should not be the flashing red light of saying, okay, it's here. Instead, we need more support than just a single detection. And so this shows that as you go down the branches of this tree, you are really collecting more samples, looking for more positives. And it's giving you, as you go down this tree, increased confidence that this is not a one-off, that this is in fact a true positive detection. And that's really important for both managers and the geneticists. And as you can see, you can then also divide this up by your risk tolerance in general. Next slide. I can see I'm about out of time, so I'm going to really speed up here. And so a couple new technologies that are coming down the pipe. One is an aquatic invasive species biochip, which has several aquatic invasive species on it. Having this sort of tool will allow you not to have a crystal ball of what's going to be coming next into our system. This has 50 of the most invasive species that invade aquatic ecosystems on it. And so you can use this to start doing more high throughput detections very sensitively, very cost effectively. Next. Also, environmental RNA is another area that's really up and coming as a complement to environmental DNA. And although I don't have time to go into it about its advantages and applications, one thing that I want you to note is that it may enable better spatial and temporal resolution in terms of what a positive detection is and validating that, especially if we use it in conjunction with the environmental DNA. Next. And then lastly, a major new area that I'm particularly excited about is the Monterey Bay Aquarium Research Institute has these tools for aquatic environments called environmental sample processors. They can go out in, and they even have little submarine type torpedo shaped things that they can move themselves through the water system to go wherever you want to collect environmental DNA in the water. And they can be put out for months at a time. You can store these filters for later processing in the lab. But even more cool in my mind, because I'm a geneticist, is that you can conduct the molecular tests inside this machine itself. So you can do qPCR and other things, and then they remotely send you the signal to shore if you've got a positive detection of whatever organism you were looking for. Next. We have developed very rapid genetic identification methods based off of CRISPR technology that allows us to distinguish different smelt species. And we are now working at doing the same for invasive species, such as Nutria and the Dracaena mussels. Next. And just as of yesterday, I started meeting with the MBARI folks to collect some samples so that we are going to start a collaboration about combining Sherlock onto those ESP tools. Okay, thank you so much for your time.
Thank you so much, Melinda. That was awesome. Lots of updates regarding eDNA. We don't have time for questions right now, but I mean, I'm super curious about how, maybe major challenges or opportunities for eDNA use in the Delta, but maybe I'll put that in the chat and we can talk about it later. But right now we're gonna move on to our kind of last chunk of the day. And this is really our more interactive session. So I'm gonna queue up a presentation for Rachel and then just pass off the mic right now.
Great. Thanks, Rachel, and thanks to everyone who has spoken today. It's been really great to get all this information about early detection and rapid response to inform some of the work that we have been doing. So as Rachel said, my name is Rachel Wigginton, and I am the Senior Environmental Scientist at the Delta Conservancy, and I'm also the facilitator of the Delta Interagency Invasive Species Coordination Team. And I'm really excited to talk to you all today about some of the work the DISC team has been doing to develop an early detection and rapid response framework and coordination table for the Delta. I wanted to take a moment up top to acknowledge the DISC team, and particularly the EDRR working group and Nick Rasmussen from the Department of Water Resources, who has individually contributed a lot of effort into the drafts that we're going to be discussing today. Next slide, please. Here's a quick outline of what I'm going to be covering. First, I'll do a quick reminder on who the DISC team is before going over the purpose and scope of our EDRR work. And then finally give a brief introduction to the draft EDRR framework and coordination table that we've developed. Next slide, please. As Laurel mentioned at the top of the program today, the DISC team was formed in 2013 with the aim of fostering communication and collaboration among California groups that were working on invasive species management, specifically in the Delta. The team is co-chaired by the Delta Conservancy, the Delta Stewardship Council, the California Department of Fish and Wildlife, and the California Department of Water Resources. And we are seeking to meet the aim of the DISC team through a few major avenues. Next slide, please. First, we work to exchange information, and this happens in several ways, including our quarterly team meetings, as well as the biennial Delta Invasive Species Symposium that you all are participating in today, and was planned by a working group of DISC team members. Next slide, please. The DISC team seeks to leverage funding for important work related to invasive species. An example of this is shown in the image on this slide of a restoration planting that's an experiment looking to exclude invaders from these restoration sites. And this project was developed by two DISC team members, the Department of Water Resources and UC Davis researchers. Next slide, please. Finally, the DISC team focuses on research and management needs for addressing invasive species in the Delta region. A major feature of this work is a research needs table, which was last updated in 2018. The previously discussed restoration planting experiment was actually identified through the process of updating that research needs table. And another major theme that emerged during the last update of our research needs table was a need for more capacity to support EDRR in our region, one of the most invaded estuaries on the planet. Next slide, please. So we as the DISC team asked ourselves how our group could increase the effectiveness of EDRR in the Delta. And here you'll see a very simplified version of the EDRR process that you all have been seeing a lot throughout presentations today, cycling through from preparedness to rapid response. Next slide, please. And to make all the parts of the EDRR process function well, we need adequate information provided to authoritative decision makers in a standardized and timely manner, as well as effective coordination among actors. As we all know, the Delta is a really complex region when it comes to natural resource management, and there are already many EDRR resources and programs on the landscape, as we've talked about in today's symposium. However, a lot of these efforts are siloed by species or legal jurisdiction, which can cause a slowed response when a novel invader comes on the scene. Next slide, please. In response to this, we started developing our draft EDRR framework and coordination table, which has a scope of the aquatic, wildlands, and habitats of the legal delta. We also hope to coordinate with efforts both up and down estuary, some of which Laurel highlighted at the top of today's symposium. Next slide, please. Now I'm going to briefly introduce the elements and sub-elements of the draft EDRR framework and coordination table for the Delta. This draft framework is strongly based on the national EDRR framework put forward by the Department of the Interior in 2016 and the 2020 special issue of Biological Invasions, which was focused on EDRR, both of which have been discussed several times throughout the symposium, but were highlighted particularly by Stas in our first session. We see this as really setting the stage for a more comprehensive EDRR plan for individual species, like what Teresa described this morning for the Colombia Basin. Next, please. The first element of our framework is preparedness. The sub-elements here are horizon scanning, planning, and research. The draft framework describes how preparedness feeds directly into all the other elements and sub-elements of the framework. Next, please. The next element is early detection. Here, the major sub-elements that our working group has defined are training and monitoring, detection and reporting, and communication and outreach, which I feel like we've learned a lot about, or at least I have personally, in today's symposium. Next, please. The third element of the framework is rapid assessment. The sub-elements here are risk screening and risk assessment, where risk screening is a super fast process based on information that's already been gathered, and risk assessment is a more of an in-depth and often defined by policy type of process. We also include risk management and risk communication under this element. Next, please. The final framework element is rapid response. The sub-elements here are coordination and leadership, containment and quarantine, the actual treatment and eradication of the invasive species, and finally, monitoring, adaptive management, and documentation after a hypothetically successful eradication. Next, please. I wanted to make a brief note here that prevention activities exist outside of the EDRR process as our group has defined it, but prevention and EDRR efforts should really support and inform one another. Next slide, please. Each element in the framework is given a definition, an illustrative example to show how the element might be applied, and lists key gaps in the Delta's EDRR resources related to that element or sub-element. Next slide, please. So let's take an example of the sub-element of horizon scanning. Next, please. First, we have the sub-element definition. We define horizon scanning as the systematic examination of potential threats, the activities undertaken to prioritize invasive species of concern, and the means to address their introduction and spread. Each of these definitions in the draft framework is accompanied by a source citation, so any potential users of the framework know how we arrived at those definitions. Next, please. An illustrative example of horizon scanning is the ecological risk screening summaries generated by the U.S. Fish and Wildlife Service. These summaries provide a rapid evaluation of a species' potential invasiveness, as well as quick ways to determine if the species are most likely or less likely to cause damage if they were actually to invade a region outside of their native range. Next slide, please. Finally, we put forward some key gaps. In this case, our horizon scanning ability. We felt like we needed to highlight a need for improved watch list resources so we can increase cross-training of monitoring teams already out on the landscape in the Delta to have them look for particularly troublesome species. Next slide, please. So to me, the next logical question is, how do we choose the key gaps that we chose to highlight in the EDRR framework? Next, please. First, we worked with a working group of the DISC team members that met about every month from December 2020 to August 2021. This group used their expert knowledge of the Delta and existing EDRR-related activities happening in the region. Next, please. We augmented this data with some desktop research. So in other words, we spent quite a lot of time digging around in the web resources and contacts across multiple groups in the Delta. Next, please. And we consolidated these data into the draft Delta EDRR coordination table. Next, please. The coordination table is organized around different EDRR resources, organizes the different EDRR resources around 4 different EDRR elements, which are, again, preparedness, early detection, rapid assessment, and rapid response. Pulling together this data allowed us to make some assessments about key gaps. Next, please. For example, we learned that we have many resources in the Delta to support preparedness and early detection, but far fewer resources for rapid assessment and rapid response. Next, please. Similarly, we found that there were many agencies involved in current EDRR activities in the Delta, but our ability to rapidly assess and respond to new invasions is concentrated in a smaller number of actors. Next slide, please. So now I'm going to quickly provide an overview of the structure of the draft Delta EDRR coordination table. Next, please. So the current EDRR table is currently in an Excel format, and you all received a copy of that Excel sheet in the email with the agenda for today's symposium. And each row in the Excel table is a unique Delta EDRR resource. So the different columns that allow you to sort this data include a unique identifier for each EDRR resource, the organization in charge of it, the program within that organization that runs this resource, the organizational mission, and then the website, just for general information. Next, please. We then categorized each of the EDRR resources, next, please, by the different EDRR framework elements and sub-elements to which they applied. This allows future users to be able to sort the EDRR coordination table based on the element of the EDRR framework that they're hoping to implement. Next, please. And finally, we're working on including columns where we categorize the EDRR resources by the taxonomic group to which they apply and the jurisdiction in which we are actually able to act on those resources. Next, please. So a few next steps. First is to gather information on the draft documents, which we're going to start doing here in just a moment. And then the DISC team will come back together in 2022, both the whole team and the EDRR working group, to start to incorporate some of the feedback from the symposium into these drafts. And then we are planning to engage resource managers in 2022 in a workshop where we start to really get into the nitty-gritty of how we would apply and implement an EDRR framework for the Delta. I know this was a very brief overview of all the work that we've been doing, but I hope that it provides you enough context for those documents that you were sent before to make a little bit more sense and for you to start beginning to provide us information about how we can better organize this data to help you all in your work most effectively. Next slide. And with that, I'll just say thank you and turn it back over to Rachel.
All right, thank you so much, Rachel. Appreciate the overview. And as Rachel mentioned, the table, the actual Excel file and the Word document with the coordination framework are both in all of your emails. We're having issues connecting it to the chat now, but those were sent out. We look forward to feedback. We are also now going to ask for some feedback specifically on, oops, sorry, today's workshop. So let me get this all pulled up. I'm hoping that folks can see a Mentimeter that we have. So for folks that have used Mentimeter before, if it doesn't look right on the screen, let me know, but I think I got it. On your computer or on your phone, if you go to menti.com, that's menti.com, you'll be able to type in a code that's at the top of the screen. I'll read it a couple of times in a second, but we're going to go through getting some feedback from you all in real time, which is very exciting. So if folks can go on to menti.com on your phone or on your computer and type in the code 1647. Now I just got it covered on my computer. I can't read the rest of it because my Zoom information is blocking it.
I'll butt in here and say 16473753.
Awesome, thank you. And as you can see, folks are adding in the responses to this first question is, do you work in the Delta or elsewhere? We're trying to understand kind of and it's only a few options in the Delta, San Francisco Bay, elsewhere in California, or out of state. We'll give folks just another minute or two, or actually more like 20 seconds. And then in a second, I will advance to the next slide and it'll automatically take you to the next one. So if a couple more folks want to log in. All right, so it looks like most of the attendees today are work in the Delta, but we have some folks from elsewhere in California and the Bay or out of state. Maybe that's Teresa. Okay, so the next question is, how many years have you been working on invasive species? 0 to 5 years, 5 to 10 years, 10 to 15, or more than 15 years. I already have quite a few folks that have spent a lot of time working on invasive species management, which is awesome. All right, I'm going to move to the next question. It looks like we have most in the 0 to 5 range, but quite a few with a lot of experience here. Next is specifically, how familiar are you with early detection and rapid response? So options are, the symposium was your introduction to EDRR. Maybe you've read about it. Maybe you've done some EDRR work, or maybe you are an expert. I see that we have quite a few. But it seems like most are falling in the range of, in the middle here of read about it or done some EDRR work. But also some folks that have just been exposed from this symposium, which is also really exciting. All right, we'll jump into the next one. Have you had time to review the draft EDRR framework and table that Rachel just presented? This helps us understand kind of where folks are at leading up to the symposium. So we have a few folks, awesome, that have read it all. Some have read most of it, some have skimmed it, and some haven't looked at it, but I'm interpreting that to say haven't looked at it, but you're about to open it up after this and read it all in detail. Okay, that's awesome. This is good to know, good to know. It's, yeah, like I said, it's been circulated via email, but we'll be in touch, making sure that everybody has access to that framework table and the report itself, or the outline. But that's great to know. Awesome. And thank you for the few of you that have read it all. Okay, so next one. How do you think you would use the draft early detection and rapid response framework and coordination table in your work? And this is like an open-ended response option, so you can type in multiple responses if you'd like. So far, already seeking funding. Some will not use this. To advocate for it and its funding, yeah, having a framework in hand is part of the, a big step to be able to communicate its value. Justifying funding, networking and funding, coordination, finding partners in work, seeking funding, better designing experimental plans, helping other agencies, definitely funding, a big component. I'm just scrolling here. All right, excellent. Oh, we're seeing more answers pop in. Strengthening state systems, collaboration opportunities. These are awesome suggestions. Okay, fantastic. Showing off, that's awesome. I think that's maybe SciSac. Yeah, coordination, collaboration, fantastic. So we have a few more of these open-ended responses, but really, really helpful to see this feedback. The next one is, are there any actors? And I know this one might be we'll give a short window for this because I think most folks haven't read the report, but are there any actors missing from the framework and coordination table? So if you haven't had a chance to look at it, or just not sure, no worries. But if you have, and you have some folks that entities, agencies in mind that should be included that we overlooked, we're here for it. Taking like silence in this as a maybe a pass on this one. Okay, yeah, RCDs. More federal or at least stronger connections with federal partners. These are helpful. Okay, like 3 more seconds. And then I'll jump to the next question. All right, so this one is, are there any concepts missing? You can think of like elements, concepts missing from the EDRR framework and coordination table. Again, this one's a little bit tough to answer if you haven't had time to really digest all of that, but you know, if anyone has ideas, concepts, elements, key steps, maybe. Maybe this will be a nice follow-up later. Citizen science. All right. Yep. That's a great one to ensure that we have in there. This one's a word cloud, so it's there's only one suggestion. So. All right, I'll move on. Oh, we got implementation funding. That's a good one. Citizen science. Yeah, make sure those components are both or elements are included in there. All right, I'm going to hop to the next one. What data sources or monitoring could be helpful for EDRR in the Delta, either existing or needed? You know, what are important monitoring, important data sources that we have, and what are ones that come to mind that we should pursue? More eDNA. All right. I'm sure there are other ideas. iNaturalist, like a online or a app for reporting detections. Spatial information. Makes me think of some of the talks we had towards the end of the day today and the importance of that. Yeah, more about eDNA, remote sensing, again, community science. Explore modeling to the fullest extent possible. Yeah. And then other methods for monitoring low density species aside from eDNA. That's a question to include indigenous approaches. Again, like spatial and satellite information or data, iNaturalist. Oh yeah, sharing IDs. Re-implement the CDFA statewide aquatic vegetation surveys. All right, yeah, some of these suggestions might be to reinstate or provide additional funding to existing programs. So super great suggestions. I apologize, but we're going to scoot to the next one. What mechanisms, either this could be legislative, regulatory, or reports, could help support EDRR implementation? Again, some of these are a little tough right now, to answer right now, but getting some really great suggestions so far. And for my co-coordinators, I can't see the chat at the moment. So if there are things that are coming up, just let me know. Okay, so some of the mechanisms, again, that can support EDRR implementation: funding pot, targeted briefings, dedicated state funding again, Senate oversight hearing, maybe streamlining the incident command protocol, a coordinator at the regional and state level, or state level, apologies, legislator meetings, statewide alerts when new invaders are detected, the Infrastructure Investment Act. Someone's supporting the coordinator idea. These are fantastic. All right, like 3 more seconds, then I'm going to hop to the next one. Legislation to secure long-term funding. Yeah, we've heard a little bit about those funding pieces that have been critical to the presentation today. Okay, next one. This is just there's not going to be any answers that pop up on the screen. And I'm about to pass it off to our next kind of brief speaker. Elizabeth, I think, is going to wrap things up, unless Rachel has a couple words first. But if you want to be involved in more EDRR conversations or with the DISC team moving forward, please type in your email here. We get the data on the back end, but it's not going to share any results on the screen. I'm going to stop sharing, but this is still open. I'm not going to close this yet. So if you want to go ahead, Elizabeth, mic's yours.
Okay, thanks, Rachel. And so I was just asked to provide a short wrap up today as a way of finishing out the symposium. We've heard a lot of great talks and definitely some common themes among them. So we certainly heard that EDRR requires system level thinking and innovation. We need to think ahead, including thinking about climate change effects, think beyond individual species, and be creative in our approaches. And three things that came to mind as I was watching all of these talks are the need for preparation, prioritization, and partnerships. Preparation, first of all, meaning use all the available information that's out there to inform our EDRR efforts. That could include things like models, that includes data. That means there's a need for data sharing among agencies or using citizen science apps like people were just mentioning a minute ago. We really need to have structures in place for monitoring and for response when an invasive species is found. And that means things like flexible funding and an ability to work across agency jurisdictions. It's also important to build capacity for the people who will need to respond. So we heard a really good example of that with the Columbia River Partnership and its quagga zebra mussel exercises. And then for prioritization, that means prioritization of both potential invasive species and locations to survey for them. We heard about a number of different risk assessment tools that are available. There are the formal ones like ICAT, or ones developed more locally like Orange County CNPS's method. Prioritization can also mean thinking about the biology of a particular species in relation to physical factors like CDFW does to prioritize sites for quagga and zebra mussel monitoring. Again, the need to use available data and identify data gaps that are needed to improve prioritization. And then finally, the importance of partnerships. This can mean partnerships between agencies with an MOU or some other formal agreement so that they can respond together and work in a cooperative way when a new species of concern does arrive. Building contacts with potential partners. Maybe those are volunteer organizations to expand the range of monitoring and reporting. Having relationships with landowners can be very important. And then the need for partnerships between researchers and managers, especially we heard about that with eDNA and some of the other new technologies that are coming up, to think about how new technology can be integrated into current efforts and also how it works within the structure of the regulations we have that a lot of agencies need to work within. And so the EDRR framework and coordination table are going to be attempting to address many of these issues. So if you have not had a chance to look at those, I encourage you to do that at some point in the next couple of weeks, and do send us your comments and we will incorporate them in 2022 going forward. And with that, I'd like to end by thanking all of our speakers for their great presentations. Thank you to all of you who attended today. Please do make your colleagues and friends aware of the recording when it's available. We'll send a link out for that so that more people can watch this who couldn't attend today. And thank you to the technical staff who handle all of the logistics of today. And so with that, thank you for attending our biannual Delta Invasive Species Symposium. And I'm going to adjourn the meeting here.